exciting research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogoriense Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO. Quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration, and each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer, and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the third largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO, and has 3 international centers, namely Asia-Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. LIPI options, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum established in 1817, is the third largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO, and has 3 international centers, namely Asia Pacific Centers for Ecohydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders.
Good morning. Assalamualaikum warahmatullahi wabarakatuh. Our plenary speakers, moderators, and all of distinguished participants. It's a great pleasure for me to welcome all of you on the second day of International Symposium on Applied Chemistry 2020. This day, we have a plenary session too, and also two parallel sessions at noon. But before we start this day, I want to tell all of you the several announcements to all of the participants and also our keynote speakers and moderators. First, please all of you have a stable internet connections. Make sure that computer has a working camera and audio device. In our symposium, the host will automatically mute the audio all of the participants, and the chat room is only available for delivering questions to the ongoing discussion in the session, requesting for changing or moving to the other breakout room in the parallel session, and announcement that delivered by organizing committees. Dear keynote speakers and also all of participants, don't forget to fulfill the list of attendees that we share the link in the chat rooms. And please properly rename your Zoom ID according to your participation category to set you up to the correct breakout room in the parallel sessions. For the participants that want to asking question, please state your name of institutions and also the panelist name that you want to deliver the questions. Okay, ladies and gentlemen, we will start the plenary session too. In the plenary session, we have three speakers. First, Niles Prakas Nirmal, PhD, from Mahidol University, Thailand. Associate Professor Dr. Nur Huda, from University Malaysia Sabah, Malaysia. Professor Nina Artanti, PhD, from Research Center for Chemistry, LIPI. This plenary session will moderate by Dr. Renat Andri Frediansyah, a researcher from Research Unit for Natural Product Technology, LIPI. Dr. Renat Andri Frediansyah graduated from his PhD in pharmaceutics from University of Tübingen, Germany, during 2007 until 2020. During 2017 until 2020. His master's degree got from Chulalongkorn Graduate Institute of Thailand during 2013 until 2015. As a young scientist, Dr. Andri got many awards. First, ASEAN Youth Summit Fellowship in National University of Singapore, and also Best Student Research Award with the Japan Society for Environmental Chemistry, and also many awards. Well, ladies and gentlemen, I will give time to our moderators, Dr. Renat Andri Frediansyah, to conduct this plenary session.
Thank you, Dr. Marisa Angelina, for your very generous introduction. Ladies and gentlemen, welcome to the second plenary session of the 6th International Conference on Applied Chemistry, focusing on natural medicine and also nutraceutical. I am Andri and I will be your moderator for this plenary session. The session has 3 keynote speakers from 3 different countries, first Thailand, and then Malaysia, and our country, Indonesia. Before we begin, I would like to give information to all participants. Before Dr. Nirmal starts his talk, I would like to give an overview of his background. Dr. Nirmal is foreign expert at the Institute of Nutrition from Mahidol University, Salaya, Bangkok, Thailand. He achieved his PhD in food chemistry from Prince of Songkhla University, Thailand in 2011. He was also a team leader for Wildbread in Brisbane, Australia back in 2018 to 2019. Dr. Nirmal also research fellow at the University of Queensland, Australia from 2015 up to 2018. His research interest is bioactive from plant and their derivative as alternative sources of food and food additive. He also interested in developing functional food product, food nanotechnology, seafood or agri-waste utilization, and also nanoscience. Dr. Nirmal also actively published his research in the top international journal, and currently he has a more than 35 journal. And ladies and gentlemen, please welcome Dr. Nilesh Prakash Nirmal. The time is yours and you have maximum 40 minutes to deliver your talk from now. Dr. Nirmal? Yes. Doctor, are you hear me?
Hello.
Yeah.
Hello.
The
Am I
time is
audible?
yours.
Am I
Yes.
audible?
Yes. Now, yes.
Yeah. Thank you. Thank you, Andrew, for your great introduction. And I would like to thanks the LIPI organizing committee for inviting me to this session. Can you see my presentation?
Not
Angie?
yet.
Yeah, can you see
Not
my
yet.
presentation? Yeah, okay.
Now, yes. Yeah, now, yes.
Okay.
You can start. Yes.
Yeah. Well, I would like to thanks organizing committee for inviting me to give a talk on this occasion of 6th International Symposium on Applied Chemistry, focusing on natural medicine and nutraceutical. So today I'm going to talk on a biological activity of fish protein hydrolysis. So I choose this topic because we know all over the world there are a lot of people now who is moving from like animal proteins to the seafood proteins, mostly like aquaculture or fisheries production. Okay, so the fish, they are moving towards more towards fish protein because of delicacy and easily digestible protein. So what we're gonna see in this topic today is the introduction, and then sources for fish protein hydrolysis, then production, purification and identification of any peptides, and their various biological activities. Further, we will see the structure activity relationship of fish protein hydrolysis, and additionally, we will see what are the challenges for large scale production and commercialization of this fish protein hydrolysis, and the research gap in nutraceutical applications, and finally conclusion on this topic. So in introduction, as I told you that nowadays many peoples are moving towards the fish sources, seafood sources for the protein, then these fish and fish products are well known because of their easily digestible protein, and the global fish production increases everywhere, including aquaculture and fisheries like the catching fish from seas, and the aquaculture like producing fish or others seafood in aquaculture like that. So we can see in table 1 the world fisheries and aquaculture production, which is produced and presented by the FAO in during 2020. From table we can see the aquaculture and world fishery production increases steadily from 2006 until 2018. We can see all the even the sea capture or aquaculture production is keep on increasing because of demand from the consumers. They demand more and more like uh, more and more fish product because of uh, highly easily available protein and nutrients and everything compared to other protein sources. So yeah, here we can see in 2018 the total production reached until 179 million ton, among which almost 156 million ton is consumed directly for human consumption. So that means it's all directly utilized for um, human food, but the rest of is like use around 22 million ton is used for other sources like making feed product or animal feed product or other things. So in this, almost 27 of the total fish capture is unutilized because of loss of quality or storage problem or loss of quality. So somehow this 27% from this 170 million ton fish, they are unutilized. That means they cannot sell in the market. So they have to either dump into the sea, or they have to find the alternative source for that. So, and then remaining harvested fish go through the processing plant, where only 20 to 50% portion of the fish is utilized as an edible product, and the remaining parts, like 80 to 50%, again discarded as a byproduct or leftover. So what this leftover or byproduct contain? This leftover or byproduct contain like skin, head, viscera, bones, scales, and fins. Because obviously we just like fillets and protein part. But how about this waste? This is completely waste. So some portion is used for the animal feed production, or if someone, they don't have any technology or anything, they just dump in the land or sea anywhere. So it causes like environmental hazardness. So what we can use this either unutilized fish or the fish processing waste. So these byproduct actually contain 58% of protein and 90% of fat and some content of minerals. So it has the potential to become reutilized. So we can reuse this waste, we can make some process, and yeah, we can utilize either for, again, for animal feed production or for human consumption again. So what we plan here, like these waste or waste product can be utilized to produce the fish protein hydrolysate. So fish protein hydrolysate, so far there are a lot of research that it shows the fish protein hydrolysate containing active peptide exhibits various biological activities. So now we will see what are the sources. So as I told you, sources, it can be fish flesh or muscle, like we usually eat directly, but we can use that flesh or muscle to produce a fish protein hydrolysate also. The second is fish skin, and then other byproduct, which include like scale, viscera, head, and fins. So fish muscle or fish flesh contain the myofibril protein or sarcoplasmic protein. Then fish skin is high in collagen. Similarly, this scale and head, they also have other proteins, including collagen and other protein sources. So we can use these protein to produce a fish protein hydrolysate. So for this, what are the production technology? How we can produce the fish protein hydrolysate from all these raw materials? So raw materials, it can be either whatever available for you, or you want to utilize the waste, or you want to make from flesh or anything. So there are 4 different methods so far, like people are using mostly for the production of fish protein hydrolysate. The first, enzymatic hydrolysis. So enzymatic hydrolysis contain, obviously as the name shows, enzymatic. That means they use enzymes for degradation of proteins. So enzymatic, so there are different kinds of enzymatic, like endopeptidase, exopeptidase, and all that. And now we can get in the market like alkalase, Protamax, Nutrase, and there are a lot of varieties of proteins, commercial enzymes available, which you can use in the process of enzymatic hydrolysis. The second is chemical hydrolysis. Chemical hydrolysis, the very first, they use like acid hydrolysis or alkaline hydrolysis. but these are like really hazardous to operators and environmental also. So chemical hydrolysis, you can use either alkali or acids. And the third is auto hydrolysis. So what is auto hydrolysis? Auto hydrolysis is using the enzyme which are already present with the raw material. Every organism or everything, they have a proteolytic enzyme. They just give the active condition including pH and temperature, so these enzymes get activated, and these proteolytic enzymes they degraded the protein itself in the same part. And the fourth one is the fermentation hydrolysis. So fermentation, as it says, fermentation. So fermentation, they use a different bacteria to ferment the medium, and the medium contains either your fishery waste or any like whatever you want to get the hydrolyzed. So there is like again you have to get the active pH and some mineral contents of the media so that the bacteria can grow and produce. Again, there is a proteolytic enzyme or a protease enzyme, variety of protease enzyme depend on the bacteria source you use. They produce all these enzymes, and they when they produce the enzyme, these enzymes cleave the raw material, which is like very now complex, but they utilize as their growth time increase, they cleave and they produce the small fragment like peptides from the proteins. And then so this is the four way that we can produce the hydrolyzed from protein, large molecule protein to a hydrolyzed. So what they generally do is we collect the fish source, and then we homogenize raw material with the water, adjust the pH and temperature as per the requirement. If you are going to use like enzymes from external or autolysis or fermentation, so we need these pH and temperature active so that these enzymes can be active and it can completely degrade the protein. Then addition of enzymes and hydrolysis. Then we have to add the enzymes, and then there is a particular time depend on the procedure and everything. We have to allow to completely hydrolyze the protein into the hydrolyzed. Once you make sure that now the hydrolysis is done, then we can deactivate enzyme. We can deactivate enzyme by changing the temperature or by changing the pH. Once the deactivation is done, centrifuge hold the contain and then collect the fractions, collect the filtrate, and that filtrate is your fish protein hydrolyzed. Actually, it's a protein hydrolyzed, but because we use the fish sources, so we call it as a fish protein hydrolyzed. So what is the next step after this? The next step contains the purification and identification. So now it is depend on your research interest that whether you want to go for purified or you just want to use like a crude, whatever you prepare fish protein hydrolyzed. But now, because due to a lot of competitions, many researchers, they wanted to purify it, they wanted to identify what exactly the peptide sequence which are giving a particular biological activity. So that is the interest of scientists. So for that they go for the purification. So purification obviously follows the chromatographic techniques which control filtration, ultrafiltration, gel filtration, size exclusion, affinity. And there is one more I would like to add, like any enzyme bioactivity guided assay. Okay, so we can use that technique also to purify your peptide. So all these chromatographic techniques like size exclusion, which can exclude the smaller size first and retain the large molecule. So this is what affinity, like based on the positive and negative charge. Gel filtration also work on the same principles. So once you separate your peptide or the fraction of your peptides, and then once you get those fractions of peptides, you we gonna screen the activity, biological activity. Suppose you are looking for antioxidant activity, so we just check the antioxidant activity of each fractions, and wherever you get the highest antioxidant activity, we use that fraction for further identification or further high purification. So for that, we go with the identification, go with the LCMS and MALDI-TOF. LCMS is the liquid chromatography coupled with mass spectrometry. MALDI-TOF is the metric assisted laser deposition, ionization, time of flight technique. So these techniques are work on the like ionization of the molecules, and then they detect that molecule and molecular weight and everything. So here you can get actually the molecular weight, also the peptides, and the sequence also you can get. There are another techniques like peptide sequence and all, you can do that also. So the next step, position and content of that amino acids in the peptide sequence. Additionally, the bioactivity of protein hydrolysis depend on various conditions used during the hydrolysis. As I talk about the production, we can see that during the hydrolysis, like the types of enzyme, as I told you earlier, exopeptidase, endopeptidase, or the enzyme to the substrate ratio, pH, temperature, and time of hydrolysis. All these factors define the final product or the final product composition of your hydrolyzer. Whether it can be a small peptides or it can be a large peptide is almost depend on your time of hydrolysis and the purification technique you use and everything. So once you get this fish protein hydrolyzer, this exhibit for biological activity screening. So far they have reported like majority of the activities including antioxidant, antimicrobial, antihypertensive, calcium binding ability, anti-diabetic, immunomodulation, and anti-proliferation activity or anti-cancers activity. So this is what the overall, what fish protein hydrolyzed can give to our body, or how it can be a benefit, or why it can be used as a nutraceutical applications. There are a lot of like some miscellaneous activities also with this we will see. Now, antioxidant activity. So these days, almost everyone now, they want some antioxidant. Wherever they are just looking on, oh, I'm looking for food which has high antioxidant, or we are screening for the phenolic compound from plant source or this source, which should have high antioxidants and microbial and other activity. So this antioxidant activity is very important actually, very, very important, because in normal living being, we actually the oxidation happen in every part of cells, or even in human body or plant also. Oxidation is the normal process of the life cycle. But sometimes the over oxidation can create a deadly or notorious reactive oxygen species, which is such as like superoxide anion radicals, hydroxide radical, hydrogen peroxide, and singlet oxygen. So once these excess free radical form, they are really unstable and very reactive. They can damage your cell protein, including nucleic acids, even lipid. They cause very serious damage to the cell and subsequently the human body. So that's why we need antioxidant. We need actual antioxidant. That's why many people, they are looking for a food like keep healthy lifestyle and trying to get the food which can give you like antioxidants. So we mostly they go for like a fruits which produce the most phytochemicals, which are known antioxidant, highly antioxidant. So another things I would like to tell you how these ROS or reactive oxygen species they form actually usually as a oxidation process. But the one important factor is stress, which increase all these things. which increase the number of reactive oxygen phase and inflammation, and it act like a precursor for all these things. Yeah, so now what these antioxidants do, they are the stable active chemical compounds. They are capable to intercept, combat, and prevent or reduce the reactive oxygen species. So how these antioxidant molecule they do? They do by different process. So usually there are three mechanisms, like scavenging free radicals, like these free radicals, or quenching the singlet oxygen, and third is a chelating metal ion. So these are the three well-known mechanisms carried out by antioxidant compound. So now you might have surprised, what is why chelating metal ion? Chelating meaning the having a complex or make this metal ion unavailable for the reactions. So why the metal ions? Because some oxidation reactions is occur through metal ion in through phantom reactions, and they increase the oxide superoxide radicals. So that increase like reactive oxygen species. So far, there are different analytical and cellular method used to assess the antioxidant capacity of fish protein hydrolyzed. So what are the analytical methods? The analytical methods consist of like free radical scavenging activity like DPPH, superhydroxide, then hydroxyl radical scavenging activity, then metal scavenging activity, then ORAC. ORAC is the well-known analytical method. Plus, there is like lipid peroxidation inhibition activity. These are the analytical activities which you can do in the flask on a chemical basis. And there are some cellular methods. So nowadays, many people, they prefer cellular antioxidant because it resembles more to human body or cellular activity. So cellular antioxidant activity consists of like antioxidant activity in cells, then lipid peroxidation inhibition in human red blood cell. Another one is inhibition of oxidative DNA damage or protein damage in the cells. So these act, yeah, obviously everyone knows protein is the long chain or amino sequence of amino acid. Protein consists of various amino acids, and then it is formed as a protein. So these amino acid also has the side chain active molecules which are involved in various activity. So the most reactive amino acids are like acidic side chain, aspartate, and glutamate, and sulfur containing side chains including cysteine and methionine, and aromatic side chains. Additionally, shorter that means low molecular weight peptide generally show higher antioxidant capacity compared to larger peptide or polypeptide. This is obvious because small molecular weight peptide are easy for access to the reactive oxygen molecules, or it can easily penetrate within the cells and it can easily react. However, the large polypeptide, because of large change, it has some adverse reaction here and there, and the active molecule is not available to counteract or interact with the radical ions. And the aromatic side chains, they are well known metal chelating because of phenolic ring. So they have ability to chelate with the metal ions. Now the next one is the antimicrobial activity. So why the antimicrobial activity also? Yesterday we have seen Professor Chaudhary. He have mentioned about the multi-drug resistance bugs. So we know how these bugs, they are deadly bugs. So researchers all around the fields or everyone, they are trying to find a natural source, including plant, herbs, and everything, getting phenolic compound to try to find some bioactive compound which can kill these microorganisms which are really deadly to human beings. So there are other sources like this, which we call like antimicrobial peptide. So this fish protein hydrolyzed has a tendency to work against this bad microorganism, and it has the ability to kill those microorganisms which are hazardous to human being. So those bioactive peptide which can act as an antimicrobial peptide, what they have, so they have basically hydrophobic amino acid content at the C terminal position. Okay, how these act? Because these hydrophobic amino acid residue, they are actively interact with the negative charge surface and phospholipid membrane of the bacteria, result in disturbing cell wall and killing them. As we know, I hope everyone knows the bacterial cell wall and everything, it has the negative charge and phospholipid membrane. So mostly these small protein hydrolyzate or small peptides, they are able to interact with this negative charge, and they are able to, after interaction, they can disrupt the cell wall and then cell lysis. This is how they kill these bad bugs. That's why this is a very important activity which can be shown by fish protein hydrolyzation. Another one is angiotensin 1 converting enzyme. So now hypertension. You know, hypertension is the leading cause of heart disease. So why angiotensin 1 converting enzyme? What is the role of this enzyme? So what this enzyme do? First, I would like to tell you this is a non-specific dipeptidyl carboxypeptide present in renin-angiotensin system. So what this renin-angiotensin system do? This system regulate arterial. Therefore, to inhibit this ACE enzyme is commonly used as a first line of therapeutic treatment to control the hypertension. So this protein hydrolyzate, which can shows the ACE inhibitory activity, they are mostly, these inhibitory activity depend on amino acid composition and the molecular weight of the peptide. Okay, so the presence of hydrophobic amino acid, again here hydrophobic amino acid, arginine and valine at the C terminal. So please note that what we have found, most of the biological activities of peptides, they show the activity, and these peptides contain either hydrophobic or any particular amino acid at the C terminal, carbon terminal, which increase the ACE inhibitory potential of the peptide. There are some in vivo study as well. So it is not just in vitro, but there are some in vivo study uh, where fish protein hydrolyzate given to the spontaneously hypertensive rat. The content was like 600 milligram per kg body weight, which shows significant lower systolic and diastolic blood pressure. So it is not only the flash analysis or analytical process that it can inhibit the ACE inhibitor activity, but there are some mice study or rat study which shows the successfully they can lower the hypertension in hypertensive rat. Now, another activity is calcium binding activity. Now why this calcium binding activity here? Okay, as we know, calcium is an essential element for all living organisms. Calcium play a vital role in many cellular mechanisms. Even they are helpful for bone formation and then everything. So this is really important for well function of the body. So calcium deficiency is the worldwide problem owing to insufficient intake or decreasing the solubility. So actually, this is the major problem. Usually after age of 30, our calcium is, our body is not able to absorb the calcium from whatever the food we have eaten or whatever the tablet we eat, our body is not absorb that completely calcium. So there is a problem for the solubility of calcium or availability, bioavailability of calcium in the body. So that's how this fish protein hydrolyzed play a role here. Calcium ion bind with amino nitrogen and carbonyl oxygen atom of fish protein hydrolyzed. So once this calcium, like the major amino acid responsible for strongest calcium binding properties of peptides are aspartate and glutamate residues. So when we have this complex, this fish protein hydrolyzate and calcium complex, it has the great solubility, and it's increased the bioavailability for our body. It not just like go in the waste or we cannot utilize. It increase the bioavailability to our body because it is chelated with this fish protein hydrolyzase. So this complex fish protein hydrolyzed and calcium complex could be an empty source as a calcium supplement. The next one is dipeptidyl. Is the dipeptidyl inhibitory activity. Again, now this enzyme involved in diabetes, type 2 diabetes mellitus. Again, yesterday, Professor Chaudhary have talked about the diabetes also. And he have explained many compounds and how this, why it is important to find the bioactive molecule from natural source which can control all these disease. So this is now another like from natural protein hydrolyzed from fish waste or uh, any fish sources. So this DPP4 inactivate incretin, which is involved in insulin secretion. So reduced insulin secretion cause type 2 diabetes. Therefore, this is very important to control this enzyme. So the lifestyle, pathogens, and foreign antigens. So to modulating immune action, immunity of the body. So how like exercise, eating habit, many other things which can boost the immune activity. The immune response triggered by bioactive peptides considered to be one of the form of immunoregulation. So here it is like bioactive peptide, how they boost this immunoregulation. So what these peptides do, they upregulate the immune function by different route. Upregulation of immune activity, such as increase of TGF beta. They increase the transforming growth factor beta, and also splenic NK, natural killer cell toxicity, and macrophage phagocytosis, and level of serum increase all these activities of all these uh, cells and the activity of molecules so that immune system get boosted. By doing this, so down regulate the including TNF alpha, that means tumor necrosis factor alpha, then interleukin 6 and 1 beta, then nitric oxide, PGE2, prostaglandin E2 production by deactivating NO synthase and COX mechanism. So so far we saw all these fish protein hydrolyzed or peptide, they are having activity. In each activity their mechanism of action is different for every biological activity. The next one is anti-cancers or proliferative activity. Yesterday we have seen our keynote speaker Dr. Ambara, right? Yeah, Dr. Ambara, they have he have talk about the breast cancer. He have talk about the breast cancer, and he have shown like how his synthetic probe can help to recover the cancer patient and how it can reduce the anti-cancer activity and all that thing. So the same, like as I already told you, there are work differently in every reaction. So what this cancer is defined as abnormal growth and proliferation of cell in the body. Therefore, controlling the initiation or promotion of cell proliferation is an important task for treating any malignant tumor. So this is the major step, like how to control. We have to control this malignation or initiation or promotion of cell at the start itself. So here, these peptides, they work very well. They shows this anti-proliferative activity through cell toxicity, cell cytotoxicity, and other mechanism like apoptosis and blockage of nutrient. So you can see this is a natural produced from a natural source and a very like body friendly, you know. It may not have any more inflammation or something to your body because it is prepared from natural source, and it can work through apoptosis and blockage of nutrients. So yes, it is promising future research for this fish protein hydrolyzed. Now, so these are the mostly known activity. Now other reported activities. So there are other reports, people, as I told you before, researchers, scientists, they are still looking as these peptides, they are has a versatile biological activity. What else they can do? This is the eagerness of scientists. What else they can do? So they try for different. So far, antithrombic activity, anticoagulant, then anti-obesity effect, anti-fatigue, anti-atherosclerosis, anti-aging, anti-glycation, cardioprotective, neuroprotective, hypocholesturemic, and wound healing activity. Yes, all these activities, this fish protein hydrolyzed has a capacity or has a tendency to act in all these disease condition or to improve the health of human being. Now, so what importance, how all this fish protein hydrolyzed shows this biological activity? The important is structure activity relationship of fish protein hydrolyzed. As I told you, that the most important thing, the structural characteristic of fish protein hydrolyzed, which includes molecular size, chain length, amino acid composition, and sequence, and their functional group. These are the most important thing, because these things, like mostly the amino acids and their functional group, they are involved in the all these biological activities. And obviously the size, because smaller size, it facilitate more interactions, it increase the interaction with particular area or disease or reactive species, such as like this structural activity of fish protein hydrolyzase is different for different biological activity. The most bioactive peptide consists of hydrophobic amino acid in peptide sequence, basically at C terminal, carboxy terminal. So this is what they have concluded, that the most active, it means it can show various biological activity. This protein hydrolyzate has hydrophobic amino acid at the C terminal. Nevertheless, molecular size, chain length, and functional side chain of amino acid also influence the structural activity for the particular bioactivity. Now, after we seen that this fish protein hydrolyzase is so much promising, it's like it's a really good medicine for most of the disease. It has ability to cover, and it has ability to improve um, many things. So what is the problem then? Why there is not many products in the market? Actually, we have some product. There are some product, but those products are like fish protein, collagen hydrolyzed, but in a particular range. It is not any single peptide or anything. So what are the challenges? Challenges are the continuous availability of sufficient raw material. As yes, this is the very important. To produce anything, we need the raw material, continuous raw material. Then only your any production system can be successful. The second thing is difficulty in the production of specific quality of peptide. Like like I said, I want this particular peptide sequence. So it is quite difficult to produce in a large scale. So far, whatever the laboratory scale production is successful, but real practice or pilot scale study is quite different from the lab preparation. Reproducibility and consistency in batch to batch production, that is also another challenge. And the next one is the stability of bioactive peptide during production and storage. This is the biggest one. And the third, for commercialization, cost of the raw material, production process, and marketing. So these are the things while they go for the large scale production and commercialization. Next, nutraceutical application. Still, as I told you, there are very few products, that too from the Japan. They are selling some gelatin hydrolyzed or protein hydrolyzed, but they have something like only like 2000 kd up peptides or something like that. It is not any isolated peptide or something. So what is the nutraceutical research gap is the toxicological study of particular peptide sequence. Then pharmacokinetics, the one we have to know application or nutraceutical application, we have to study all this toxicological study, pharmacokinetic study, and then long-term effect of consumption. We have to study that also. So if we consume every day, like long, like 6 months, 1 year, what will be the effect on our body? Because we will taking this as a supplement or nutraceutical added. So what is the effect? Then large number of clinical trials for particular disease condition. The next, safety, environmental and legal consideration of raw material and final product. Yeah, legal consideration is very important. That's how the regulatory harmonization all around the world is needed to become acceptable nutraceutical product all around the world. The conclusion. So this is the wonderful area and growing area. Due to its various biological activities, researchers are still interested. They are still utilizing new free sources, various different parts, using different processes. They're trying to get the different easy, fast, cheap processes to get the higher production and many things. There are a lot of scope for this. And then sources like unutilized fish. Like there are some small fish. Sometimes when fishers, they go for the catch of fish, they get the fish which is people like we don't eat. So they just throw it or dump it. But they can use these unutilized fish. Then they can get the fish muscle, fish skin and other byproduct. This protein hydrolyzation show different in vitro and in vivo biological activity. The gap between commercial production and nutraceutical application of fish protein should be overcome. The biological activity of fish protein hydrolyzed open the door for nutraceutical industry. These are the references, actually just small references I'm showing here. There are a lot of references, and moreover, you can get from our publications also. There are a lot of, you can just search my name. I work on shrimp processing waste also. So a lot of different and various application and waste utilization, which is published in Trends in Food Science and Technology this year. Yeah. So Thank you. Thank you very much, everyone, for your attention and listening me. However,
Okay.
yeah,
Yeah.
Andre, I would like to
Yes.
request you, if I have another 5 minutes or 10 minutes, I would like to promote my
Mhm.
startup, my business here. Can you
Yeah.
allow me
We
to do
still
that?
have. Yeah. We still have around 6 minutes for you. Maybe you can
Okay,
start
6 minutes.
from now.
Wonderful.
Yeah.
Yeah. Thank you for allowing me to show my business. So I'm happy to tell everyone and announce that I have started the business with Hillberry Life Sciences. This Hillberry Life Science is the production, everything is in India. And what we are doing, we are a manufacturer of herbal extract, essential oil, and dietary supplement, capsule formulation. So all the materials are Indian origin, and we have the state of art of manufacturing facility with GMP, halal, FDA certification, then ISO, HACCP, GMP certification, and everything. So as per your need, we can make all the supplement, including immune booster, then for bone osteoporosis, and a lot. So what our services offer, we offer the best manufacturing services. We offer a contract research and development services and contract manufacturing organization, a capsule manufacturing processes, which include product design, formulation, packaging solution, stability studies, regulatory support, which is important also, depend on the country, and then investigation of source and availability of all raw materials. Not only that, we provide independent and collaborative technology development to provide tailor-made solution for customer specific demand. So we we do have our products in the market, like you can see, like Blackmore and all those, the nature products. They have their own product for each particular disease. So we also have that. And not only that, we can produce capsules or do the stability study or regulatory study for any product which you would like. So if you are interested for any quotes and order, please contact me on this email, nirmalnp21@gmail.com. And here you can see my mobile number. This is the Thailand code 66, and my mobile number 8345 72612. So here I would like to offer one more chance for those who are interested to do a business. Those who is interested to do business with us, I'm welcoming them. Please contact me. The business, like, you have to get the industry contact from your country or all around the world, and let's start the business, commission based business. So you can, this is a really good opportunity. If anyone interested, please contact me. And here is my email address and my mobile number. If you not get, then you can call, contact the Abdi, and you can get my contact from him. Thank you very much. Thank you very much. And thank you, Andre, and thank you, organizing committee, for having me here. Thank you.
Thank you. Thank you, Dr. Nimal, for your very great talk. Your talk about biological activity of fish protein hydrosolate for human health. That's very useful for everyone. And also, good luck for your startup. I hope everyone could join with you.
Yes,
If
yes.
you
Thank
have
you very
any
much.
question later, yeah, if you have any question for the audience, you can ask in the chat, and we will discuss in the end. And
Sure.
because the bad connection in Malaysia, so we wait for Professor Huda until he could join with us in Zoom meeting.
Okay.
Therefore, we're going for the next keynote speaker from Indonesia.
Okay.
The
Yeah.
speaker of this plenary session is from Indonesia. Yeah, I told you. Are you ready from Professor Nina Artanti? Are you there? Just to make
Okay.
sure first.
Yep. Okay.
Yes. Okay.
You can hear me?
Yes.
No,
Okay. She is a professor, Dr. Nina Artanti, and she will deliver her talk about Indonesian biodiversity of source of functional food. Before Professor Artanti starts her speech, I would like to introduce her professional background. Presently, she is a the research professor at Research Center for Chemistry, the Indonesian Institute of Sciences, or LIPI, Indonesia. She achieved her doctoral degree from Ehime University, Japan, in Applied Bioscience and Biotechnology. In 2007, she was also fellow holder for a short course conducted by JICA Bioindustry Group Training in Japan. She has also received a 2-month research fellow from participating in training on antioxidant bioassay and hepatoprotective at Zhejiang University, Guangzhou, China, with Professor Pan Yuangjiang and also Professor Lu Jia as a part of LIPI Zhejiang University in traditional medicine collaboration. Currently, her research interest is broadening from the biological activity of natural product and also functional food. Professor Artanti actively published her finding as a part of dissemination in more than 20 papers in Scopus, and she was also appointed as a reviewer in. Ladies and gentlemen, please welcome Professor Dr. Nina Artanti. The time is yours, and you have about 40 minutes to deliver your talk.
Thank you very much, moderator. Okay. The share screen is okay?
Yeah, we can see your slide now.
Thank you very much for this opportunity. Assalamualaikum warahmatullahi wabarakatuh. Today I will give a talk on Indonesian biodiversity as a source of function.
Professor?
Yes.
Could you increase?
We have 17,000 islands when only 6,000 is inhabited, and for the sea territorial is also large. So then the population is about 274 million people. As this large country, we also known as one of the world mega biodiversity country. It's comprised of 1,500 species of algae, 80,000 fungal species, 595 species of lichen, 2,197 fern species, and many flora of the world flora. And in terms of terrestrial, we are probably number 2 after Brazil, but we also have the marine. That's why we have maybe the biggest biodiversity stores. And we usually already know about this biodiversity we use to find new drugs and everything. But of course, it's also a good source for functional food. Is it okay, or because?
Sorry for your
sorry
for
okay.
internet. Yeah,
Right.
true.
I thought that is a close connection. Okay. And so, what is I also think that this biodiversity should be also explored and be used for the development of functional food. And what is a functional food? There are many definitions about functional food, but they all correlate to each other. That is a food that will be more than just us to have the nutrition, but also to have effect on health uh, and should be consumed as a food. So it's probably differ from the nutraceutical of something that then can consume as a tablet or something. It should be good and have a taste nice, and so but so it will be a bit different from the development of the bioactive compound as we use it for drugs, the tuber crops. There is many kind of several type of tubers in the uh, it's still not utilized very well, like arrowwood, gembili, gembolo, suet, porang, iles, uwi, taro, pers, and kana. Because in major agriculture only usually just use cassava and sweet potato and also potato now, but the other is just less used. So it's actually a good source of uh, starch like inulin and also anthocyanin, glucomannan, which has low glycemic index, which is good for diabetic people. So we should use we explore the other tubers in Indonesia to use it as a source of functional food. I think our colleagues in Gunung Kidul and also in Subang also try to make the functional food based on these underutilized tubers. Maybe if I can go back, sorry, this is the ones that suak is the same family of the bunga bangkai, and this is I think the black potatoes, and I also there's some is talas this one, and in biotech before we already tried to make the propagation with the tissue culture, and this the cassava, but we increase the beta carotene to get the more beta carotene to make it yellow, so it has a higher beta carotene content. I forgot this one. What's this? Okay. Uh, so this is also some underutilized vegetables. This is kucai, and there's this pegagan in this kelor. I think the this labu, I don't know the English name, is more still not to utilize, and this something like grass that's rarely used. But this is some medicinal tradition of some Indonesian underutilized vegetable that's have the medicinal or traditional use. So it can be developed into making the functional food, like for the allium, it's for antihypertensive. Centella, that's activity that's I think is also known as a part of cosmetics, used for cosmetic, besides of the good in also for memory enhancements. And daun moringa is calor, good for the content of protein, and besides it's good for antihypertensive, diabetes, and also heart disease. This underutilized vegetable will rich source of polyphenol and ascorbic acid and carotenoid. Okay. And we in Indonesia we eat a lot of rice, and most of us I think usually just eat the white rice. But then actually there's many variety of rice in Indonesia, and like the black rice, and some is a red rice. Banana is divided into the genome A, B, B, and SS. Banana fruit is useful for is alternative of staple food raw material of processed food with high carbohydrate and potassium that had potential for non-communicable disease. Okay. And one of Indonesian biodiversity that also high in carotenoids is the Pandanus conoides. It's an endemic plant to be efficacious. Extract of froxin contain of alkaloids that can be used instead of morphine for pain relief. In addition, froxin extraction also contain a substance that is able to active the pancreas gland that can be used as an anti-diabetic drug to stimulate insulin. There are at least 347 species snakes in Indonesia. Traditionally, people use the extract of whaling snake at Bungarus candidus to lower blood pressure. Like the diversity of flowering plants, Indonesia also has a wealth of bees. Traditionally, bee venom may be used for arthritis, lumbago, and skin disease. The bee venom contains apamine, melittin, phospholipase, hyaluronidase that suppress the nervous system, stimulate the liver, and adrenaline glands. And For the marine, from the marine, we have a lot of sea cucumber species that have a lot of bioactive compound is long been used as food or traditional in Asian countries, and the price is actually expensive. And actually, there is a 350 sea cucumber species from Indonesian water have been recorded, and at least 26 with economic value. And like Sticobus hermani, Talentota ananas, Talentota anax, and Holoturia folia, and Actinopiga mauritiana as most highly valued species. These organisms are potentially source of high value added compounds with therapeutic properties such as triterpene glycoside, carotenoids, bioactive peptides, vitamins, minerals, fatty acids, collagen, gelatin, chondroitin sulfate, and amino acids. In the recent years, health benefit effect of sea cucumber have been validated through scientific research, healing, neuroprotective, anti-tumor, anticoagulant, antimicrobial, and antioxidant. This functional material lead to potential development of food and biomedicine industries. And Indonesia also known with a lot of fermented food. And we know there's is good for the function. H3 has many characteristics that are benefit for functional food processing. It's a non-reducing sugar that is soluble in water, has half of sweetness of sucrose, is stable in acidic condition at high temperature, is resistant against the browning reaction and the non-caloric sweetener. The FH3 has several health benefits, enhancing the uptake of calcium, zinc, and magnesium in the small and large intestine of rats and humans, inhibits the hydroxy reaction of bile acid to provide secondary form, which is a risk factor for colon cancer. Therefore, the FH3 is expected to be a new prebiotic candidate. So is the now I will talk about the some that's research in the of functional food in Research Center for Chemistry. This is a natural folic acid. It's work for Miss Agustin that I think some of you already hear heard her presentation yesterday, and there's another one today related to these studies. So we make the natural folic acid based on the like tempeh from soybean and also from uh, green bean and from the uh, nixtamal of corn and as well as some fermented vegetable using lactic acid bacteria, and then we make it as a source of natural folic acid by identifying using the LCMS to make sure that we got the certain amount of folic acid and use as a mistletoe species. That's because it's continuing my research when we are in trying to isolate what the because mistletoe is usually used for traditional medicine in for cancer therapy, alternative medicine maybe you call it. And we found we test that from her antioxidant and anti-cancer, and we found when we mix the cakar ayam soto and make tea herbal tea preparation, the activity is better in the mixture. So it is a good, and it's because it's already like people used drink this one. So, but for commercialization, maybe we need to do like the proper and activity testing. Although for the C, as well as zinc and its ionophores. And innate immune defense also increased by thymus extract, probiotics, flavonoid, and sulfated polysaccharides. About sulfated polysaccharide, I haven't make the presentation, but uh, this year we are continuing from last year project, the Incinas. Dr. Sofa also make the uh, biscuit for cookies from uh, algae, the marine algae caulerpa, and now we're going to mix in with the microalgae is from to because we found it's have a immunomodulatory activity, and one of the is sulfated polysaccharide that they found isolate from the caulerpa. So it's also like we found that oxidative stress can be reduced by vitamin C, and vitamin D is can be from the fruit and also fermented fruit, and also the inflammatory reaction of some botanical and probiotics. And hemostatic immune system also increased by vitamin C, flavonoid, and polyphenol. So what the because this COVID-19, so
Professor,
yeah,
Professor, you have
yeah,
still 5 minutes left.
yeah.
Okay.
Maybe I finished before that is okay. Isn't it? We got the Miss Yati Mariati involved in making the guava fruit fermented with kombucha that help to increase our immune because it will be produce some uh, the fermentation could be produce some acid that it's not formed if we just make the guava juice. And then for Miss Hani Mulyani and yeah, Satyani also try to make the herbs what call it marmalade that contain, but it's not just the herbs, they also fermented it with lactic acid bacteria, and then make it to the make it a marmalade that can be used for the bread spread or to drink. So it can be both to do both, and this hopefully it also increase the immune system for people, so we can avoid the COVID-19. I think I finished my presentation, and hopefully that the functional food that we develop from our Indonesian biodiversity that also hopefully will keep our immune high, so we don't get COVID, can be together with the there is industry or the we can collaborate with the uh, small medium price enterprises that but this also need good collaboration, and then hopefully this can be used for people. This now we already make it and for distribute it to try for people to try it, but not the commercial production. Okay, thank you very much.
Oh, thank you so much for Professor Artanti. Very nice talk related our biodiversity. This remind us that we have so many sources that need to be explored, especially for human health, from nutraceutical and also natural products. Thanks again of herbal tea. I hope you could market the tea very soon. And for the next speakers, so we have Professor Dr. Nurul Huda. Now he is here or not?
Yes, I'm here.
Could you hear me?
Yes, Dr. Andri, I can hear you.
Okay, I stopped it. All right.
Hello?
Oh, sorry. I don't know. Okay.
Sir, we cannot see you.
Yeah, I cannot
Okay.
the video. So sorry, so my internet connection is very poor, so I'm not able to join early with you all. So I try to many.
Penang, Malaysia. Professor Huda achieved his doctoral degree from the Food Science program at the University of Kebangsaan Malaysia in 2001, master degree from Food Science program in Bogor Agriculture University, Indonesia, and his bachelor also from Indonesia from University of Bung Hatta. Professor Huda received various excellent awards, including High Impact Publication Award and Excellent Service Award from University Science Malaysia in 2012. And also he has High Impact Publication Award from the same university in 2013, Service Award by University of Sultan Zainal Abidin, and recently in 2019 he got Research Exhibition Award from University of Sabah, Malaysia. And ladies and gentlemen, please welcome Professor Dr. Nurul Huda. The time is yours.
Okay, thank you, Dr. Adi, for nice introduction about me. First, I want to ask my sorry to all of the organizing committee and also to the keynote speaker, Dr. Nima, and also Professor Anita. I expect your presentation must be a very important thing, but I'm not able to join with you to follow your presentation. So actually, the title of my presentation is. Can I share my slide by myself, Dr. Aldi?
Yes, you can. If you could share your slide. I didn't see up to now.
Yeah, I think better if I share my slide by myself, so
Okay.
I can freely by myself. So can I share by myself my slide? Can I try now? Yeah.
Yeah, please
Okay.
try.
Okay. Can you see my slide now?
Still ongoing. I will wait for that. Yes, now I can see.
Yeah, we can see now.
Yeah, you can start from now.
Okay, you can see my slide now. So the title of my presentation is Exploring Application of Psychology as a Wound Healing. Maybe some information will be introduced by Dr. Niles Normal because we are come from a similar field. It's more fish processing technology. Until I'm the close friend of your Professor Benjakul, and also maybe Dr. Nina. So my presentation is focused on the fish collagen. Because maybe for Dr. Nima is quite, I expect is talking with quite rank of the fish protein hydrolysis. So first, let me introduce my place, my university. My university is located in the north part of the Borneo, the north part of the Borneo, University Malaysia Sabah. So our university consists of 3 campus. The main campus is belong in Kota Kinabalu. The International Finance Faculty is located in the one island, Labuan Island, and the Sustainable Agriculture Faculty is located in Sandakan. Is the area of my current campus. So actually, our university is very beautiful university. So I invite once the profit is we can hold this and we describe to the environment, to the environment such. Then I focus on the wood healing. Collagen as one of the byproduct to healing property. So maybe some of us, we still remember is around 2 years ago. The New Zealand doctor tried to treat the bone torrefied explosion with the fish skin. There is, I think it's 3 to 4 years ago. Is quite viral in animal how they use directly the fish skin to treat the bone victim. But then with this idea come to the surface how to produce how to study is more detail about the potential of this fish skin. Actually, the fish skin is considered as a fish collagen because the main component of the fish skin is belong to collagen. And then to treat the what call it burn victim look like not ethics, not nice. Yeah? If our body is covered with the fish skin, then we need to think the proper technology to deliver this potential of fish skin or fish collagen as raw material for the wound healing process. This is the idea activity wound healing potential from the byproduct of the tropical fish. About the phase of the wound explain with the wound problem is that need to three phase for the as wound healing material. Chitin, shrimp, and also alginate. And so sea cucumber, I think as mentioned by Nina previously, this cucumber also the potential raw material for wound healing material. And also catfish and so chitosan. It's a list of the natural product that we can use as wound healing material besides the collagen. So this is the evidence that how the some natural component is able to act to accelerate the wound healing process if we treat with some potential bioactive component. So we can see from the report of the Parfin Pirzania, they use aloe vera, honey, and milk ointment for the secondary burn impact. And then you can see from this picture, this one is without any, this one is control group. One is belong to control group. Control group, the animal is treated by saline solution only. And then the group 2 is treated with with ulcer, simple sulfadiazine is a commonly treatment for the burn people. And then the group 3 is belong to the honey, milk, and aloe vera ointment 5%. And then we can see that treatment with honey, milk, and aloe vera is able to to close the wound is much faster, is much good, better than the control one, and also compare with the origin and sulfonyl with 3%. So this one also another evidence from the curcumin, from curcumin conjunctate with hyaluronic acid. So hyaluronic acid we can extract from the heat of the chicken, from the red part of the heat of the chicken we can extract the hyaluronic acid. So we can see this one from the graph by Dr. Sharma. We can see the untreated one. The untreated one is the one closure is quite low. It's only around 25%. But if we treat with the animal with curcumin, and then the wound closure is much significant higher increase. And so if we if we treat with hyaluronic acid, if we combine together curcumin and hyaluronic acid, and then the wound closure is almost 100%. So the next evidence also from the fish oil. So meaning we can use different kind of the product to as a wound healing material, wound healing agent. This one from the oil from the fish head, and then you can see from this result. So if the control group normally there is belong to the control treatment with only consist saline solution, and then compare with Kikaflora, the commercial treatment, and then HSO is a hair oil. Is a fish oil extracted from the fish head, and then we can see that product use that treatment using the hair oil, the animal the wound closure is more faster compared than control one, compared than Kikaflora. So you can see from this graph. So within the 11 days, so the totally 100% of the wound is totally closed. If we treat, if the animal treat with the fish head oil, comparison control one, and also comparison commercial Kikaflora. So and then let me see the collagen, and let me see the collagen because they mentioned because they mentioned previously that actually the skin is consists collagen. So the similar structure with our skin. Our skin also consists collagen. So actually, according to collagen is actually from the Greek word is a glue and to produce. So actually, the collagen firstly explore because it's a component tissue when we boil and to produce glue. And after that, some development of the collagen science and technology. And so far, the 29 distinct collagen type have been characterized. So collagen is considered a different type. So far, the 29 collagen type is characterized. Then all of the collagens, 29 collagen display are typical triple helix structure. So what is the trademark of the collagen is a collagen. This collagen is consists of the proline and hydroxyproline. So normally the structure of the alpha chain of the collagen is consists a sequence of the glycine, proline and hydroxyproline. So why normally in order to determine whether the material come with high amount of the collagen, we can conduct analysis based on the content of the proline and hydroxyproline, what we call as amino acid. So normally raw material come with high proline and hydroxyproline. It also come with good property of the collagen, can form the gel. This one also, however, they mentioned to you 29 type of the collagen is characterized, but the only 9 from the 29 is only what the is upon 9 is most belong to the most common collagen type. Is the 9 most collagen type is available in this raw material. This one just inform to participant about the collagen. Meaning, if you talk about collagen, is not only collagen. There are 29 type of collagen is available, and then we need to understand the structure, the characteristic of each collagen in detail to know more knowledge about collagen. So actually, this one will happen if we experience with the animal or people experience with the wound. What we say wound accident. So actually, if the healthy collagen is come with the triple matrix, and this one is a healthy collagen. But if we experience with wound, some damage is take place. And sometimes it takes place, and sometimes it takes place in the triple helix. And then with addition of the collagen from outside, small molecule of the collagen, it will be able to faster, quicker copy of the damaged collagen. This is one of the scenario is proposed by scientists, how the collagen peptide is able to accelerate the wound healing process due to the small peptide of the collagen from the outside. This thing, as I mentioned previously, we have a lot of fish here with 70% is consistent marine, a lot of fish species available there, and then we need to explore together the potential of the fish species as a wound healing material. So we can extract this collagen from the marine fish, of the marine fish, especially from the byproduct. Byproduct you may consider from the skin, from the bone, or from the scale, and to produce a collagen. So when we produce a collagen, and then we need to hydrolysis a collagen. We need to go to next step to produce a collagen hydrolysis. And then when we produce a collagen hydrolysis, maybe with a different property, which depend to the process, depend to the enzyme applied, depend to the acid we use during the extraction. And then we can apply, we can test the produced collagen hydrolysis as a wound healing agent, whether in different form, whether in the sponge form, cream form, hydrogel, or spray. So far there are less knowledge and less report on the potential of the fish collagen as a wound healing material. There are some report on jellyfish, Alaska polar, tilapia, salmon, haddock. But all of the report show the acceleration of the wound healing rate by using this kind of the collagen hydrolysis. But you can see from the list of fish, most of the fish is not belong to tropical fish. most the fish is belong to temperate fish. It means consider maybe if we need to explore the potential of our tropical fish as a raw material to produce the collagen hydrolysis with wound healing properties. So actually, this collagen hydrolysis from the fish collagen is a good chemo attractive for granular tissue formation. Promote according to previous report, promote cell proliferation and attachment, a good biocompatibility and biodegradability, and maintain tissue hemostasis as long as antigenicity. And a good best material for the potential dressing is according to the previous report. But I don't see any report from the tropical fish for this time being. Most of the raw materials used by the previous researchers is belong to the temperate fish. So maybe probably between the temperate fish and the tropical fish, there are some different in terms of the properties. So it's a way, it's a one, it's a extraction method that we can apply. It's a one of the methods that we can apply to extract the protein fish collagen hydrolysis. So by fish prey product, whether from skin, bone, or scale, and we need to conduct for pre-treatment to produce the acid soluble collagen. We need to use remove the non-collagenous material. So we need to reduce the S content by using the DT. We also need to reduce the fat content by using the butyl alcohol if the fish skin come with high fat content. And then we produce the acid soluble collagen. So acid soluble collagen, we call it acid soluble collagen, and then we conduct the salting out, and then we produce the acid soluble collagen dry form of the acid collagen. But during the production of acid collagen, there are some pellets, precipitate. So the precipitate we can extract again. We can extract the precipitate by using the enzyme to produce the peptide soluble collagen. And then we freeze dry. And then they produce the soluble collagen, whether from the acid or peptide, and then we need to hydrolysis to produce a small peptide. To produce, whether we can apply any enzyme, whether collagenase, papain, alkaline, potassium. So we can use many type of enzyme. And then we can see which enzyme suitable for our raw material. And then we able to produce peptides or collagen hydrolysis, and then test to we connect to conduct the characterization and test to the animal. So this is a current study by scientists. They're using, as mentioned to you, most of the fish species they use is belong to temperate fish. I don't see a report from the tropical fish for this time being. Salmonella, and also tilapia. Whether tilapia is so available in Indonesia and Malaysia and so Thailand, but this one is belong to temperate tilapia. So and then come with evidence that the collagenous from the extract from salmonella and tilapia is able to accelerate the healing rates, promote the wound healing by controlling the inflammatory reaction and increase the wound antigenesis and collagen deposition. So similar report also on jellyfish. Jellyfish improve the structure closure. It's much faster than the control one. As for Alaska polar skin. Alaska polar can enhance the wound healing activities by oral administration in animal study using the rat. And also from the unicorn little jack skin and also to Nile tilapia skin. All of the report come to with the conclusion the potential of the fish collagen as a wound healing material. But unfortunately, as mentioned previously, all of the raw material fish species used is not belong to the tropical fish. It's all belong to the temperate fish. And then it's our time now, it's our high time now to explore the potential of the bioproduct from the tropical marine fish or tropical freshwater fish as a wound healing agent. So also from the kind of collagen for the wound healing process, so you can see from this result the collagen or the animal with wound experience. So you can see from this part, the wound closure is much faster if we treat with the peptide collagen, percent soluble collagen rather than the control one. Control meaning they use a saline solution to treat the animal. To similar report from the Mandiri by using the collagen from the cuttlefish. Jadi from the cuttlefish skin and then extract the collagen hydrolysate peptide and then treat to the animal. So you can see this one is the group 1 is belong to consider the control one, group 2 and group 3. So as you can see how the cuttlefish collagen hydrolysate able to close the wound is more quickly rather compared to control group. A similar report from the shark fin, from the shark skin. Shark skin also consists of collagen to promote the impression of the wound healing. We can see similar report as reported there. A similar result so the potential of the collagen hydrolysate to treat the animal with a wound experience. This one also the similar report using the salmonella and also tilapia neurotica skin. How this collagen extract from the salmonella skin and also from tilapia skin is able to improve the healing rate of the animal if we treat with this kind of the collagen. Similar report from the Dr. Felix. So a lot of evidence, we have evidence that the ability of the collagen from marine fish as an agent from the wound healing process as accelerator during the wound healing process. So this one also from the Alaska polar. Scapola is a raw material to produce surimi. The main raw material to use the surimi production is the collagen from the cell scan of the salmon also can act as a good potential wound healing material. So this is a challenge now for us in Indonesia with a very huge amount of the fish species to explore or as a potential raw material to produce collagen peptide, collagen hydrolysate, and then to see whether the collagen peptide produced for our tropical fish able to come with a good property as a wound healing agent. So a lot of fish species now we can use such as a lizard fish, big eye snapper, or Nemepterus japonicus, screci, or maybe different. It belong to godfish. It's a godfish. All of the fish is belong to underutilized fish. Then during the surimi production, they also produce about 40 to 30% of the byproduct. So we can use the byproduct which consists skin, scale, and bone of this kind of fish as a raw material to produce a collagen hydrolysate or collagen peptides, and then we can test as a wound healing agent. So this is my current PhD student. Yeah, we just started this project last year, but due to the pandemic, our result is not come yet. So we're not able to produce any publication yet related about the potency of the tropical marine fish. So we're working with a different fish species is available in our tropical country, and then study the abilities, the characteristic of the peptide hydrolysate as a wound healing agent. But the study is on the way now. It's quite slow due to the pandemic, so we've not entered the lab really. So hopefully after the next year we can come with the evidence. So we still keep the raw materials that we use, but we try to use some potential tropical marine fish for our study. And then it's all my presentation about the potential of the fish collagen hydrolysis as a wound healing material. Okay, thank you to Dr. Adri.
Thank you very much, Professor Nurul Huda, for your nice talk. Very interesting topic related to collagen from fish sources. And now we are for the next session, which is question and answer. There are several questions in the chat room, and yeah, so all the speakers are ready today, and I will see the question in the chat room from the first one. Oh yeah, the question is for Dr. Niles Nirmal from Dodi Dharmajana. Are there any side effect of FPH as a medicinal ingredient? Maybe you can start to answer this question first. Are you hear me, Dr. Niles Nirmal?
Yeah.
Yeah.
So, well, what is the question?
Again, the question is, are there any side effect of FPH as a medicinal ingredient? From Dodi Dharmajana.
Is there any what?
Side effects.
Side effect. Yeah, side effects of
FPH. Fish protein hydrolysis. Yeah.
See, like as I told, this is from the natural source, like whatever we are eating, like sometimes fish protein hydrolysis from fish place, and sometimes from the waste product. So it has side effect, but not really harsh, and it's again depend like whether you are applying it like from outside is not, but if we take as a supplement, it's depend on the human body. It's depend on each participant or each person how they respond to the external protein source and everything. Am I answer the question? Hello?
Committee, can you show the Dodi Dharmajana? Maybe is it okay for the answer?
Hello,
or maybe I think yes. If he has more question, he will put in the chat.
Side effect as a medicinal ingredient.
Yeah. So we go to the next question and question from Sri Priyatni. According to your information about FPH, do you have the project for large scale of FPH and what application that you have done?
Yeah, I think I have reply everyone through chat box, but I can reply here again.
Yeah, because maybe everyone want to know also for the answer of this question.
So
maybe they have the same question. Yes.
Yeah, we are now in the process for applying a big project, and yes, our past work shows that ACE and DPP inhibitory activity in our laboratory study. Yeah, these are the like applications in the sense of various biological activities, but we haven't did any clinical trials or anything yet.
Okay. I think the most of question is for Dr. Niles Kumar, but I also want to ask from maybe Professor Artanti. Are you hear me? Hello, Professor Artanti.
I think she's not there. She turned off.
Yeah, maybe I will give you one question from me. What is the main challenge as a researcher in Indonesia to mine the potential of Indonesian biodiversity? The main challenge of a researcher as an Indonesian researcher.
I think the main challenge is that we have to, if we can, if we want to go to the industry, usually it's better if the industry is
Can you
doing.
increase your voice? Sorry, I couldn't
Sorry.
hear. Yes.
As a researcher, I think not just in Indonesia, if we done research and we
Yeah.
expect the industry to come to us, it's a bit difficult. But if we already have
Okay.
a talk or something, and then what they want, and we do the research for them, it will be useful because sometimes the industry know better which like functional food that will be uh, booming or something compared to us as a researcher. We just okay, we try this, we try that, but we don't really know what like expectation from the people because we that's why we are not become uh, we don't have an industry. That's why I think that's the challenge is to know what the industry want, research wants. So I think we better collaborate since the beginning, what they want,
Yes.
and we do the research. Especially now, if we get already the facility, and they because sometimes if we do research, but then we don't have the industry who will take our research, then it's just that it's just probably just become a publication. Also, we do make a patent, but probably no one will buy our patent like that. So I think because it's
They
Yeah.
will make a bridge for
So
us to make a
I would like
collaboration.
to, yeah, I would like to give
Yeah.
my opinion and my experience in this area as well.
Yes.
So when I was in Australia, I worked for the Australian native plants, herbs and fruits. So indeed, these native plants are really, really good. I mean, very high contents of vitamin C and some other phenolic compounds. So I work on a Davidson plum and other indigenous fruits, which has really high antioxidant activity and very high anthocyanin and everything. That knowledge should come to the public. So we really have to go and meet our indigenous people or these people like native people. They have
of the collagen hydrolysis. Okay, I think it's
Okay.
my answer, Dr. Adi.
Okay. So there are still numerous questions. I will read one by one. Where is it? Ahmad said. Okay. There are also.
not mean they have the same proportion of protein content or their body composition. So that make quite difficult to produce, get the consistency in the production, and get the mm, like same reproducibility or batch to batch quality of the fish protein hydrolyzer.
Okay. Thank you. We still have more questions. I hope.
With particular sequence, that is okay, but if you go for purification of that particular active peptide, the yield is really, I mean, really low, very low. And about the hydrogel, I haven't worked on hydrogel yet, but yes, it is a really good idea. And yeah, that will be really good idea, but I'm sure that it could help uh, like anti-proliferative or to kill the cancer cell.
Okay. Still have more questions for you because most
Yeah,
of people interested with your talk. And the next is Koirun Nisa from LIPI. What kind of parameters and instrument do you use to confirm that your FPH product is successfully produced?
Yes, so usually how to confirm that? So the first thing, like if you check the biological activity of your raw material and the fraction, you can see there is something like, obviously the raw material or the first fraction may not give you any result, but the fraction can give you the result. So it shows that something has happened. Something has happened to the raw material, which is the long polypeptide chain. It has degraded, and you get the some fraction. Something has happened. So next step, what you can confirm is again you can go for the that and get the fraction, and they check the biological activity of each fraction. Furthermore, you can go for the mass spectrophotometry, which can give you the molecular weight. From there, yeah, you can confirm that, oh yes, I get the hydrolyzed or the
Utilization of fish collector healing, or we need to evaluate the metal content in the sample, especially sample near mining sites. If the sample found to be contaminated with heavy metal, can we use this kind of sample?
Yes.
Professor Huda?
Thank you for Mrs. Carla. Yeah. Yes, it's very important for us to make sure that the raw material they already use for any production is belong to safe material. So if the material come from the area from the nearby to the mining area with some what expectation that some heavy metal content is quite significant, that yes, you need to conduct the heavy metal analysis first. So because we need to make sure that raw material is used for the production of the fish collagen or collagen hydrolysis is belong to safe raw material. Otherwise, maybe the raw material is not suitable, is not qualified to use to any process, not only to produce the fish collagen hydrolysate or the hydrolysate peptide, but also for normal consumption. It's very important if the area are decontaminated or nearby the mining area. It's very important to conduct the primary analysis of the heavy metal contamination. And especially according to some previous research, if the fish live around the area with heavy metal contamination, the heavy metal content in the skin is more higher than.
It's moderate, and then but it's okay because you also it's like you eat the whole fruit that's like maybe for lowering cholesterol that you like that. But we know and we do the extract, so it has the activity for the lowering cholesterol. But usually, if we because it's eat as a food, so the concentration won't be very high like that.
In terms of application, because most of Indonesian or common people don't like fishy taste and aroma of FPH, is there any way to get rid of this of flavor of FPH? Yeah.
Yes, we have a good news. There is a way that we can avoid the fishy smells, and it can make like a plain without any smells, or we can add some other flavor which can improve the aroma and everything. There is a process called debittering process. We can use that as well. Yeah. So
Thank you. Is there more question in the
yes.
chat or not? I will check again. Maybe I have a question for Professor Huda. Professor Huda, are you there?
Yes, please.
Yeah, I just want to know, is there any difference between the product, a collagen product from marine fish and freshwater, about the quality or something else? Thank you.
Okay, thank you, Dr. Adi. I think each fish come with its own property. Each fish come with unique property. So we need to study species by species. We cannot generalize all the fish species is as one category. So why? Maybe some species is potential, maybe another species is not potential. So in general, each fish species come with its own property. So we need to study species by species, and also not only because due to the different species, but if we conduct the process differently, so the result will be different. If we use a different enzyme, and then the result is also different. So why we need to specifically. So as we need to work specifically to the species and also to the specific enzyme to produce a better quality of the collagen hydrolysate. I think it's my answer, Dr. Aldi.
Thank you so much for your answer. How about from Dr. Nile's perspective about this also? The question is from collagen, right? Collagen quality from different source like marine source and also the fresh water. How about the hydrolysate, fish protein hydrolysate? also the same quality, or
Yeah,
maybe you have
as
another perspective to answer?
yeah,
Yes.
as Professor Nurul already told you that yes, every species has different compositions, so we have to study that. And obviously, as I also answered the previous questions, like the raw material is affect your final product quality. So we have to do that. So this is the basic challenge. Although
Yeah.
Professor Nurul already introduced you that collagen has a different types, but if you can able to successfully isolate or focus on one particular type, then that is the different case, you know. But
Okay.
yeah, the composition of sea and fresh water is different. Yeah, thank you.
Okay. Thank you so much. Is there any more question in the chat or not? Maybe if still not, I have a question also for. Oh yeah, we have one more question. Okay.
The third room will be discussed about food technology and functional food, which will be moderated by Anastasia Fitria Devi, PhD. And the second parallel session will be start on until 4:45 p.m. and also will be divided into 3 rooms. The first room will be discussed about pharmaceutical chemistry, pharmaceutical technology, and drug delivery system. It will be led by Faiza Mariani, Master of Science. And the second room will be discussed about analytical chemistry and chemical engineering. Will be moderated by Dilani Putri Ramadhan Tias, Master of Science. And also the analytical chemistry and chemical engineering will be moderated by Aprilia Nurtasviati, Master of Science. We hope all of the participants will be joining in your interest in each room. But in this last day, we have a closing ceremony. So we hope all of honorable speakers, moderators, and also all of participants could join our closing ceremony on the main room at around 5:45 p.m. So we will meet on the closing ceremonies, and now you can take a break time. Thank you and enjoy your conference. See you.
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you. Okay, Dr. Nimang, see you for Nina. See you
Thank you,
later.
Dr. Nurhuda.
Okay, see you. Okay.
See you.
Thank you, everyone. Bye.
Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, The cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. Best Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 we are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bam.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi, serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat, yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu dan bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya. Salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. Step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization, furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmunya Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by Infinity Science Exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta? if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmunya Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda dan mengikuti program postdoktoral saya di Delft, Belanda. Saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They managed the product. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk pemanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, The cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda dan mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019 yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by Infinity Science Exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover with positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gotong Royong, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi, serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat, yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by Infinity Science Exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Dari presentasi tersebut, saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Technology, Indonesian Institute of Sciences has been developing packaging technology for ready-to-eat foods. traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi
Tes tes. Hello? Hello?
Hello, good afternoon.
Good afternoon.
Could we start our session from now? Or we should waiting another presenter? Okay, good afternoon, ladies and gentlemen. Welcome to the parallel session of ISAC 2020. I am Osi Arutanti. It is my pleasure to be a moderator for this session. And in this session, we have 8 distinguished speakers, and each speaker has 10 minutes for presenting the material. And I will open the Q&A session after 4 speakers finish their presentation. So, on the Q&A session, if anyone has any question, please use the chat column in Zoom application by writing your name, panelist name, and your question. So, for the first speaker, I would like to invite Mr. Muhammad Zakwan from Department of Chemical Engineering, Indonesian University. He will present saturation times test of membrane to supply clean water using reverse osmosis with laser stage injection. So, Mr. Muhammad Zakwan, the time
Yes.
is yours.
Okay. Should I show my presentation or ISAC?
The committee will show your presentation.
Oh. Should I start? Because it's a recording.
Oh, okay, okay.
And it has a video. Okay. Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Zakwan, and I'm student of Chemical Engineering of University of Indonesia. And this session, I'm going to present my paper entitled "Saturation Time Characteristic of RO Membrane Scale to Clean Water Supply with Pressure State Injection." Sorry. Excuse me?
Yeah.
Oh, yeah. Okay. And this paper is corresponded by my teacher named Seti Jabismo. Next. Okay, let's move to the background. Why did I do this research? Water sources containing high salinity are not fit for consumption and being major causes of clean water scarcity. And it happened in Indonesian subdistrict Pandeglang, Banten, especially in Tanjung Lame. And reverse osmosis is a modern technology used to produce clean water with areas facing scarcity. And the technology has been globally accepted in both water treatment and desalination process. The fundamental of the technology is separating the impurities in water based on size of pores using membrane semi-permeable. But there are some problems use RO technology. They are fouling, use high pressure, and desaturation time. Membrane fouling is the main factor caused by pore clogging or absorption of solutes on the surface of the membrane, and it reduces water diffuse through the membrane. To reduce this problem, we use multi-state design by use multi-pass system to get clean water. Next. Sorry, next. Okay, move backward. move backward. Okay, again. Okay, this is the schematic of the research. The feed flows into the UF membrane to remove large particles, and this aim to reduce the RO membrane workload and extend the saturation time of the first stage of membrane. Then, from the UF, the product is pumped into the first stage RO membrane to remove small particles with high enough pressure. Furthermore, the products from the first stage are used as feed in the second stage. Here, however, because the product used as feed is clean enough, a lower pressure is sufficient. Next. Okay. The experiment is conducted in 2 steps. The first step is employ 3 attempts, which was conducted for 4 hours of each attempt. The pressure stage used for 3 attempts respectively are 6 and 3 bars, 6 and 4 bars, and 7 and 4 bars. And the second step was conducted for 72 hours using the best pressure stage in the first step. During 72 hours, the flux decrease and TDS increase were observed to analyze. Next. This is the result and discussion. In the first experiment, use of P1 6 bar and P2 3 bars report a constant value, decreasing flux. It can be seen at key P1 and key P2, and also high cell rejection and low TDS. But for the second experiment, use P1 6 bar, lower product flux axis compared to the first. Or in other words, it has a lower product than the first. Based on the theory, this is caused by concentration polarization, which causes the flux decline during the initial period of the membrane separation process. Concentration polarization increases the osmotic pressure at the membrane wall, which causes a reduction in permeate flux. In other words, higher pressure occurred to get constant flux. However, the use of P2 4 bar in the second stage gives higher product compared to the P1 3 bars. Because P2 4 bar gives higher pressure than P2 3 bars to push water for diffusion through the membrane. And last, at P1 7 bar and P2 4 bars, 3 bars. And let's move to the second result. Next. This is the table of result of the second experiment. Okay. The next step is estimate the saturation time of membrane using flux decrease and TDS increase method. So the first one, we are going to use flux decrease method. Based on figure A, flux declined from 1.5 to 1.3 liters per minute. Total decreasing flux was estimated about 13%, and the decrease of flux is caused by the accumulation of salt that prevents water diffusion through the membrane. This is due to the accumulation of dissolved solids in the membrane. So, if we assume the membrane is changed at decreased flux of 15%, saturation time of the membrane is then obtained about 80 hours at the maximum. Therefore, based on decreasing flux, the membrane is expected to be changed at the time range of 72 until 80 hours. And figure B show a lower flux was produced in the second stage compared to the first. Figure B at range of 15 until 60 hours refers constant flux. After 60 hours, the flux reduced to 1 liter per minute. This indicates the membrane saturation was significantly achieved. Total decreasing flux for 72 hours about 13%. Furthermore, the membrane is confirmed clean after decreasing flux reached 15%. And the membrane saturation time is calculated by using the equation B to generate a range of time 72 until 131 hours. So if you use this method, we are going to change the membrane for the first stage 72 until 80 hours and the second stage about 72 until 131 31 hours. So let's move to the method TDS increase.
Sorry, Mr. Jackman.
Yeah, should
you have a problem?
Yes, should I share my presentation from my laptop?
Okay.
ppm. Therefore, using the equation, the membrane is possibly cleaned after 3,000 hours more. And for the second state, figure B shows TDS increase at the second state where membrane is changed after TDS increase about 313 ppm. Therefore, the membrane saturation time is achieved at about 816 hours. And compared to the calculation between both methods, the membrane saturation time using decreased flux method delivers sufficient result compared to increased TDS. Furthermore, the decreasing flux method is more representative of the existing theory, where the membrane saturation time in the first stage appears faster than the second. So this is my conclusion. This study showed the calculation saturation time of the membrane used the flux decrease method appears more representative compared to the increased TDS, with faster saturation time achieved in the first stage than in the second stage. Using P1 6 bar and P2 3 bars, the time of membrane saturation time at the first stage demonstrate a range of time 72 until 80 hours, while the second stage about 72 until 131 hours. Maybe that's all about my presentation. Thank you, and I'm sorry for the disturbing.
Okay, thank you very much for Mr. Muhammad Zakwan for the nice talk. For the next presenter, I would like to invite Anton Prayoga, but I'm sorry to efficiency the time. So the video will be shown by the committee. So please keep your presentation file. The committee will show the video which has been sent by the participant.
Okay, so the PowerPoint presentation will show by the committee?
Yes.
Okay. Yes, please share your screen. Can I start my presentation?
Okay.
Okay.
Ms. Anton Prayoga from Chemical Engineering Indonesia of University. The time is yours.
Yes. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all participants of ISAC LIPI 2020. My name is Anton Prayoga, a student of Chemical Engineering University Indonesia. I will to present my paper with title of Design of Hybrid RNF Permanent Staging for the Supply of Clean Water in Tanjung Lame Village. With my supervisor is Professor Sudijo Bismo. World Health Organization reported in 2015.
Assalamualaikum warahmatullahi wabarakatuh.
Sorry, the record is
of ISAC LIPI 2020. Okay. I'm Anton Prayoga, student of Chemical Engineering Universitas Indonesia. I will to present my paper, the title of Design of Hybrid RONF Permanent Staging.
I'm sorry, should I present my paper live or just show the video recording?
It's okay to show a video recording.
But the voice is unmute. Voice of my video.
Yeah, it's okay. Your video will show. So
report in 2015 showed that 9% of the world population lack access to safe drinking water. In Tanjung Lame Village, Pandeglang Regency, the groundwater source containing high salt concentration and therefore are not fit for consumption. In the rainy season, the TDS of water from community wells can reach 28 and 90 ppm. In the dry season, the TDS of the water can increase due to ground evaporation. An effective method for processing clean water from salt water is the application of reverse osmosis and nanofiltration system. However, the pressure need for the conventional single-stage RO system is very high, with potential to cause
I'm sorry. There is a voice of my video recording. So should I present my presentation live or just you play my video with audio, with my audio?
Okay, please continue live.
So please share my presentation, or should I share my slide?
Yes, you can share your presentation.
Okay. Okay, I will start my presentation again. World Health Organization report that in 2015 showed that 9% of world population lack access to safe drinking water. In Tanjung Lame Village, Pandeglang Regency, the ground source containing high salt concentration and therefore are not fit for consumption. In the rainy season, the TDS of water from community wells can reach more than 2000 ppm, and in the dry season, the TDS of the water can increase due to groundwater evaporation. So, an effective method for processing clean water from saltwater wells is the application of RO filtration system. But the pressure needed for a conventional single-stage RO is very high, with potential to cause high electric demand. Several modifications to reduce the pressure, such as configured for concentrate staging, successfully decrease the pressure, although the TDS remain relatively high. So the purpose of this study is to design the permanent staging configuration. In this RONF filtration system, permanent stream on the first stage RONF act as feed on the second stage RO. So it can reduce the pressure and the TDS of clean water produced. To increase water recovery rate, nanofiltration membrane will be combined with the RO membrane at the first stage. There is a schematic diagram of this experiment. First, we collect the feed in the feed tank and feed flow to ultrafiltration membrane as pre-treatment unit. Permeate stream from UF membrane flow to the first pump. First pump push the water to the first stage RONF. The permeate of RONF, the first stage is flow to the second stage pump, and the second stage pump push the permeate stream to the second stage RO. The permeate stream from the second stage RO is collected in the clean water tank, and the rejection stream from each stage is connected to the pressure gauge and valve to control the transmembrane pressure on the membrane. The first study is effect of transmembrane pressure on the salt rejection. Salt rejection will decrease when the transmembrane pressure increases. This is caused by the variation of pressure used, which has passed its optimum pressure to separate the salt. In the case of brackish water desalination using nanofiltration membrane, divalent and multivalent ions will be filtered like magnesium or calcium, while monovalent ions like natrium, calcium will diffuse with the water molecule to the permeate flow. When the transmembrane pressure continue to increase beyond its optimum condition, the salt flow in the feed stream will be forced to diffuse through the semi-permeable membrane with a diffusion rate that exceeds the salt flow rate in the rejection stream, so that more salt will diffuse toward the permeate stream. To obtain the maximum salt rejection, the designed hybrid RO&F filtration system is operated in the first and second stage, pressure of 5 and 3 bar respectively. At that pressure, the value of salt rejection and TDS of permeate is 97.51% and 72 ppm respectively. At the pressure of the first and second stage RO&F of 6 and 3 bar, we can see the result is close to the maximum condition, around 97.06%. Okay, the next is effect of transmembrane pressure on the water recovery rate. Water recovery rate is directly proportional to the transmembrane pressure. The higher the transmembrane pressure, the more volume of water is forced to diffuse from the feed stream to the permeate stream. The presence of nanofiltration membrane in the first stage makes the water recovery rate of the first stage filtration higher than the second stage because the nanofiltration membrane contains a negatively charged hydrophilic group. The NF membrane has a higher water flux than RO membrane. The optimum pressure chosen in this study are in the first and second stage of RO and F transmembrane pressure of 6 and 3 bar respectively. At the optimum operating condition, the value of water recovery rate that can be achieved is 31%, with a flow rate of clean water produced at 1.55 liters per minute. Based on the experimental study, the optimum condition for operating the machine is specified at 12 hours per day. So the total volume product is achieved at 1,116 liters per day. It can meet the needs of clean water from 1,005 families in Tanjung Lame village. So they are the brief summary. The optimum design, the design of RO filtration system with configuration of hybrid RO and F permeate staging, comprising 2 RO membrane and 1 NF membrane on the first stage, and 6 RO membrane on the second stage, has proven to produce clean water from salt water wells at low pressure and low TDS. The optimum pressure. To obtain optimum result, the configuration operates in the first and second stage RO and F with pressure of 6 and 3 bar respectively. At the optimum salt rejection, the salt rejection and TDS are obtained at 97.06% and 85 ppm respectively. And the water recovery rate estimated at 31% and 1.55 liter per minute correspondingly. Okay, thank you.
Okay, thank you very much for Mr. Anton Prayoga for the nice talk. So, we change to the next speakers. I would like to invite Mr. Samantha Suryo from Chemical Engineering Indonesia University. He will present the degradation of 2-chlorophenol in dielectric barrier discharge reactor. So, Mr. Samantha Suryo, the time is yours.
Hello, ladies and gentlemen. My name is Samantha Suryo. Together with Febri Bazina and Professor Setijo Bismo, will present our research about degradation of 2-chlorophenol in the electric barrier discharge reactor. Several industries such as pharmaceutical, coloring, domestic, and agricultural release large amounts of toxic and dangerous pollutants into the environment. One of the pollutants is chlorophenol plasma technology, such as the BD reactor for treating waste. The process is simple, effective, low energy consumption, and do not produce byproduct. The BD reactor produces active species such as hydroxyl radical, co-radical, ozone, and hydrogen peroxide. The objective of this research is to get results from the effect of pH on the rate of degradation of chlorophenol-based waste in the DVD plasma reactor. The main components used in the DVD plasma reactors are as follows. The number 1 is power source, number 2 slide regulator, number 3 transformer, number 4 voltmeter, number 5 ampere meter, 6 neon sign transformer, 7 pump, 8 reservoir, 9 DVD plasma reactor, 10 ozonator, 11 flow meter, and 12 compressor. Experimental variables used are the independent variables is pH, which varied to 4 and 10. Dependent variable were degradation percentage, COD, radical hydroxyl produced, ozone concentration. The control variables were the initial concentration of the chlorophenol was 50 milligram per liter, degradation time was 2 hours, gas flow rate was 2.5 liter per minute, plasma torn voltage was 19 kilovolt, liquid waste flow rate was 50 milliliter per minute. And hydroxyl radical quantification. In figure 2 shows the amount of hydrogen peroxide produced at pH 10 and 4 was 0.1 and 0.00125 millimole respectively in 120 minutes. At alkaline pH, the formation of hydroxyl radical increased significantly, while at acidic pH, ozone remained in the molecular form. Therefore, the formation of hydroxyl radical was very different. This is the ozone solubility task. The ozone solubility at pH 4 was 0.221, while at pH 10 was 0.049. This occurred because at pH 4 slows the rate of ozone decomposition, therefore molecular ozone was discovered. Conversely, the rate of ozone decomposition at pH 10 increased significantly. At alkaline pH, many hydroxyl and hydroxide ions act as initiator in ozone decomposition. Here are the reactions. And this is the 2-chlorophenol degradation. At pH 10, 2-chlorophenol experienced a degradation of 79.4% to 10.4 ppm, while at pH 4, it experienced a degradation of 71% to 14 ppm. Ozonation at acidic pH was dominated by reaction with ozone molecules, whereas at alkaline pH, it was dominated by hydroxyl radicals. This was due to the higher degradation rate at alkaline pH, and furthermore, faster degradation at pH 10 occurred due to the dissociation of chlorophenol to chlorophenolate ion, which react with ozone faster than non-dissociated species. A solution with a pH of 10 makes the dissociation of 2-chlorophenol into 2-chlorophenolate ions easier. The ion formed from the dissociation has a higher reaction rate with ozone, therefore at pH 10 it was consumed at a higher rate. Likewise, at pH 4, 2-chlorophenol is more abundant in the molecular form. At pH 4, the reaction is dominated by ozone and there are only a few hydroxyl radicals, so that the 2-chlorophenol degradation yield at pH 4 is smaller than pH 10. This is the COD, chemical oxygen demand. Mineralization at pH 4 was PM for class 2 as a standard quality requirement for industrial liquid waste. However, it was discovered that this process waste has not met the Ministry of Environment Regulation number 5 of 2014. The conclusions are the degradation percentage of 2-chlorophenol is greater in pH 10 than pH 4. In alkaline condition, chlorophenol are easily to be degraded than in acidic condition. At pH 10, the yield of 2-chlorophenol degradation reached 79.4%. At pH 4, the result of degradation of 2-chlorophenol only reached 71%. The difference in degradation yield of 2-chlorophenol is also strongly influenced by reaction mechanism according to the condition of the solution. The slightly differences between pH 4 and 10 indicate that the DBD reactor can successfully degrade 2-chlorophenol compound under acidic and alkaline condition. Thank you.
Okay, thank you very much for Ms. Samantha Suryo for the nice talk. I apologize because I called you Mr. at the first. Next, I would like to invite the fourth speaker for Ms. Febri Monica Distrianti from Chemical Engineering Indonesia University. For Ms. Febri, the time is yours.
Of Indonesia majoring in chemical engineering. I'm also representing my team, Bazlina, Samantha, and Professor Setijubismo for this presentation. On this occasion, I would like to convey the result of my research entitled Comparative Study on the Degradation of 2,4-dichlorophenol and 2,5-dichlorophenol using the electric barrier discharge non-thermal plasma reactor. This presentation outline consists of 3 sections. The first is introduction, the second one is research methodology, and the third is the result and discussion. Water pollution instigated by phenolic compound from industrial waste is one of the environmental problems requiring the development of law-compliant treatments. The industries that can generate the phenolic compound known from pesticide, pharmaceutical, plywood, pulp and paper, petrochemical industries. And based on the various previous research, a number of contaminant phenolic compounds effluent, including dichlorophenol, the present in waste has been identified in several isomers, with the most common type is 2,4-dichlorophenol and 2,5-dichlorophenol. These dichloorophenols have very low biodegradability that can pose sustained risk with direct exposure, include olfactory dysfunction, skin irritation, liver and kidney failure, diabetes, and death of the aquatic life. To reduce this impact, the government issued a regulation through Peraturan Pemerintah Lingkungan Hidup number 5 years 2014. According to that, maximum content of phenolic compound in wastewater is estimated at 0.5 part per million. Although there is no specific standard for each isomers and even the types of the phenolic compound. The technology adopted as the most effective to eliminate the dichlorophenol includes the application of the electric barrier discharge non-thermal plasma reactor. This reactor implements the advanced oxidation process, rely on highly reactive active species, namely hydroxyl radicals, ozone and O radicals, and hydrogen peroxide. And the DBD reactor has the advantage of requiring only short time, low operating temperature, high energy efficiency of the chlorophenols, especially in terms of DBD plasma reactor application. Meanwhile, this research was conducted to compare the effectiveness of DBD reactors and the degradation activity on isomer 2,4 dichlorophenol and 2,5 dichlorophenol under acidic conditions and alkaline conditions. And then move to the research methodology. The experiments involve synthetic wastewater contaminated with dichlorophenol with initial concentration 50 part per million of each dichlorophenol, and for acidic conditioners using the sulfuric acid, and for the alkaline condition using sodium hydroxide. The research initiated with the preliminary test to get the best condition, which will be used as the operating condition during the degradation process. The preliminary test was conducted to quantify the hydroxyl radicals and dissolve ozone. The quantification of OH radicals was conducted by the iodometric titration with variation at flow rate, voltage, and pH. And for the dissolved ozone, was conducted using the spectrophotometric UV-Vis with ozone test kit with variation only at pH. And the wavelength used is 550 nanometer. And for the degradation process conducted with the fixed variable, as you can see on the screen, and the analysis is for the concentration of the chlorophenol using spectrophotometric UV-Vis with phenol reagent, and for 2,4 dichlorophenol using 513.8 nanometer, and then 2,5 dichlorophenol using 505.6 nanometer. And for the COD removal, also using the spectrophotometric UV-Vis and CODH with wavelength 650 nanometer. And this is the schematic diagram of the experimental setup for wastewater degradation. As you can see, there are 4 different flows covering electric current, high voltage flow, fluid flow, gas flow, and then the gas flow with continuous system that differentiate by the color. And for the configuration of the DBD reactors is coaxial pipe with cylinder inner electrodes and spiral outer electrodes. And here are the results and discussion. The first thing to discuss is the result of preliminary tests namely the quantification of hydroxyl radicals at liquid flow rate and voltage variations. The quantity of hydroxyl radicals is represented by the hydrogen concentrations and then convert to the hydroxyl radicals concentration. The faster liquid flow rate, the lower hydroxyl radicals were produced. Hence, the highest of hydroxyl radical production was obtained at 50 million milli per minute. That is because the higher contact time perfects the hydroxyl radical formation despite the relatively low circulation. And at a voltage variation, the higher higher hydroxyl radicals were produced. Hence, the highest hydroxyl radical production was obtained at 90 kilovoltage. And that is because the higher electric field intensity facilitates the higher energy generation for particles and electrons. And then the next result of quantification of hydroxyl radical and dissolved ozone at pH variations. The production of hydroxyl radicals higher when the pH of the solution is higher, because it is driven by the ozone decomposition reaction in the presence of hydrogen ion initiator. And thus, the dissolved ozone shows the opposite result. The solubility of ozone in water lower when the pH of the solution is higher. This is because due to the shorter ozone half-life and decompose into the hydroxyl radicals as I mentioned before. And this is for the main processes of degradation of the chlorophenol. As you can see, when the pH is an acidic, the lower degradation, and at the higher pH, the degradation rate of DCP is higher. This is due to the rate of this dissociation of the chlorophenol will increase as the pH of the solution increases. And then the percentage of degradation of 2,5 the chlorophenol was slightly greater than 2,4 the chlorophenol over time due to the faster polarization and dissociation depending on the atomic position, or significant in alkaline condition than in acidic condition. The final pH of degradation process is acidic due to the formation of oxalic and formic acid as the final product. And then, as you can see on the graphic, particularly during the initial 10 minutes in acidic condition, there is a surge in pH increase. And the last is for the COD removal. The treatment result for both is still resulting high final COD levels. This is indicated large number of intermediate compounds still were formed. And then the COD removal was determined to be lower compared to the DCP degradation. That is because the process had not reached the end of mineralization to become a carbon dioxide and water. For that, this is the summary for this research. First is the electric barrier discharge plasma reactor is able to degrade for both isomers of the chlorophenols after 120 minutes processes into simpler compounds effectively without a significant difference, with the final degradation percentage of 2,4-dichlorophenol and 2,5-dichlorophenol are 88.95% and 89.5 respectively, and with COD contained in the pH of the solution following the generation of acidic compounds in a final product. And here are the reference of this research. And this is the end of my presentation. Thank you for your attention.
Okay, thank you very much for Ms. Beverly. Thank you for all speakers. So next, I will try to open Q&A session. Is there any question? Okay, if no question from the audience, I would like to ask Mr. Mr. Anton Prayoga, related to the nanofilter membrane. Have you measured how many times recyclability the nanofilter membrane for this process?
I'm sorry, I can hear you clearly. Please.
Okay, sorry. Have you measured the recyclability of nanofilter membrane for this process? How many times you can use this nano membrane?
Oh, the lifetime of nanofiltration membrane.
Yes.
I'm sorry, there is out of my research. So my research are just how to design the configuration, not discuss the saturation time, caused by falling or clogging. So I don't know how many time we have to change the nanofiltration membrane.
Oh, you didn't prepare the nanofilter membrane in this experiment?
Nanofiltration membrane, we use the commercial nanofiltration membrane.
Okay. Thank you. Maybe for Mr. Muhammad Zakwan, I would like to ask, I show that here the pressure 1 and pressure 2 influence the result of this research. It's unique because become the higher the pressure, so the result also decreases. Have you measured the pressure below density and below density? Why for P1 is 6 and for P2 is 3? Or maybe you refer to the previous result? Hello? Hello? Can you hear me, Mr. Zakwan?
Yes. Sorry.
Okay. You heard my question?
Not yet because I'm from toilet.
Okay. My question is, why you choose P1 for 6 and P2 3? Have you measured below than that pressure?
Yes. Before we doing research, me and my partner Yoga, we do some we try a few pressures. So when I choose 5 bars, so when we use 5 bars in the first stage, it can be pushed water through the second stage.
So the ratio between P1 and P2 also important?
Yes, the minimum pressure used for first stage is 6 bars. So if we use 6 bar for the minimum, it can use to bring the water from first stage to second stage to be pushed by use low pressure. If we use 5 bars or lower pressure, we can use that pressure to push water through the second stage. So it can. Can you get it? Sorry.
Oh, okay. Okay, thank you for the nice answer. The next form is Febri Monica. Oh no, for Miss Samantha. As I know that also pH value will influence the produce of radical hydroxyl, and this is the same phenomenon in the photocatalytic activity. So in this case, as I know, usually in the different condition, pH 3 is the best, and for the alkali, maybe 9 or 8 is the best condition to produce the optimum radical hydroxyl. But here you use pH 4 and pH 10. Have you the reason for this choose pH number?
Okay, so I choose that pH number is because so much industry produce waste in different pH. So we want to know how if the chlorophenol how the chlorophenol affect if it is how the hydroxyl radical produced if it is at pH 4 and 10, because so many industries produce a pH at different pH.
Have you measured the different pH from below than 4 and between 4 and 10?
No, I haven't.
Okay.
I haven't do the research about it in real.
Okay, thank you. And the last for Ms. Febri Monica, it is interesting because at the same pH value, the degradation of 2.5 is better than 2.4 dichlorophenol, but for COD, 2.4 is better than 2.5. Could you explain again why it can be different?
The difference between 2.4 and 2.5?
Yeah, why the degradation 2.5 is better, but for COD, 2.4 is better.
Actually, the difference is not significant because the ozone and radical hydroxyl works synergistically. The difference, the slight difference, is because the position of the chlorophenol, the position of the chlorophenol affect the oxidation process. So, with position at para, actually the oxidation which will be more easier than the meta position. So the 2.4 is better, the greater than the 2.5.
Yeah, thank you very much. We can give applause to the all speakers for session. And next, I will continue to this second session. I would like to invite Mr. Didit Ardi Maulana from Chemical Engineering Indonesia University. Mr. Didit will present the synthesis of nanocomposite AgTiO2 for polyethylene microplastic degradation application. So, Mr. Didit, the time is yours. And please open your video.
Good afternoon, ladies and gentlemen. My name is Didit Ardi Maulana, supervised by Professor Dr. Insinyur Slamet and Muhammad Ibadur Rahman. I'm from the Department of Chemical Engineering, University of Indonesia. My paper title is Synthesis of Nanocomposite Silver Titanium for Application of Polyethylene Microplastic Degradation in Water. So the bottled drinking water has met the quality standard of the World Health Organization, so that people believe that consume bottled drinking water is safe for the health of the body. But in 2018, the State University of New York from that study, it's known that bottled drinking water sample taken from local product contain up to 4,000 microplastic particles per liter, and from the non-local drinking water sample contain up to 10,000 microplastic per liter. The type of microplastic contaminant in the water is from polypropylene by 54%, nylon 16%, polystyrene 11%, polyethylene 10%, polyester 6%, and other. The size of microplastic is around 11 to 247 micrometer. The study began from the study before by Zhao et al. 2007. The addition of 1% of titania to the polyethylene can degrade the polyethylene microplastic up to 42% under sun irradiation and 86% under UV light for 300 hours. It takes a long time. So in 2018, Ariza Tarozana et al. Trying to degrade the microplastic high-density polyethylene in the distillate water by titania doped by nitrogen under UV light irradiation for 12 hours, so the loss of microplastic mass reached 3%. The silver as a dopant working as an electron trapper. The electron trapper is due to the energy work function on the silver that greater than titania, so that the recombination rate of electron can be prevented and cannot come back to the titania. And the addition from the silver to the titania can decrease the band gap. The decreasing from the band gap can increase the absorption performance under the visible light. So the performance of photocatalytic can be improved. So in this study, we are trying to synthesize the nanocomposite silver titania. And then we characterize the silver titania by SAM EDX to analyze the final composition and surface morphology of the photocatalysis. And we are using the UV vis DRS to determine the band gap energy and light absorption power from the photocatalyst. And then we are going to degrade the microplastic. The size of the microplastic we used are 100 to 125, 125 to 150, and 150 to 250 micrometer. The concentration of 100 ppm under 2-hour irradiation. So the final result is known by the gravimetric measurement method. The right side is the photoreactor design, where there is the UV lamp, and under the bakerless is a magnetic stirrer on hot plate, and the cover for this reactor is from aluminum reflector. So the light from the UV can reflect and focus in the photoreactor. See the result and discussion. From the same ADX characterization, we can see that the titania Degussa P25 and 100% silver doped titania. We can explain that the treatment of silver dopant titania does not provide enough topological change on the surface of the catalyst. The surface morphology of the catalyst has not changed significantly, where no other aggregate are found that can interfere with the photocatalytic performance of titanium. Based on the EDX result, it found that the mass of silver metal proper on titania was sufficient by the desired percentage of 1.11% of silver titanium. Therefore, the photoacid deposition method used for composite synthesis is appropriate and give a good result. So here is the UV vis characterization. The absorbance value of the catalyst to the light at a wavelength above 400 is increased. Based on the calculation of the band gap using the Kubel-Kammel equation of 1% silver titania composite, it can be proven that the addition of silver metal dopant can reduce the band gap to 3.02 electrovolt with an optimum wavelength of 411 nanometer. The minimum bandgap energy of the semiconductor are shown. The minimum energy required by the electron to be excited from the valency band to the conduction band. The addition of the silver internia can improve the performance of the catalyst to absorb the light in the visible light area. Okay, the result of the microplastic degradation. Here is the percent degradation based on variance in the particle size. We are trying to vary the size of the microplastic particle by 3 variables. One is from 100 to 125, and then 125 to 150, and 150 to 250. We can see that in 2 hours or 120 minutes, the microplastic can be degraded till the 100% degradation. But we can see that in the microplastic size of 125 to 150, the 1% degradation can be reached by 90 minutes. And we can see that from the right side in the figure 4, the microplastic was decreased optimally in 90 minutes at the size of the microplastic is 125 to 150 micrometer. This is related to the ability of the microplastic mixing optimum in solution by the centrifugal force. The centrifugal force is directly proportional to the mass of the particle and the speed of the particle, but inversely proportional to the radius of the path. The particle speed is influenced by the speed of steering, where the steering speed in the study is made constant at 2000 rpm, and the radius of the track is also constant because the diameter of the breaker glass used is same. Therefore, the main factor affecting the centrifugal force on microplastic particle is the mass of the particle, which is caused by difference in particle size. And the conclusion from this paper is the optimum synthesis method. Photoacid deposition method used for composite synthesis is appropriate and give a good result that is performed by very simple procedure and optimum performance. The degradation performance was obtained up to 100 under UV irradiation in the presence of steering, which affected the mixing of microplastic particles in the sludge. Okay, thank you.
Thank you very much for Mr. Ridit for the nice talk. The next, I will continue to discuss second speaker, Mr. Muhammad Haris Fadli. Mr. Muhammad Haris Fadli will present microplastic pollutant degradation in water using modified TiO2 photocatalyst under UV irradiation. Mr. Muhammad Haris, the time is yours.
Good afternoon, everyone. My name is Muhammad Haris Fadli from Universitas Indonesia, and today I want to present my paper about microplastic pollutant degradation in water using modified TiO2 photocatalyst under UV radiation. The outline for this presentation is first introduction, experimental method, results, and the last is conclusion. For the introduction, nowadays we know that plastic pollutant is polluting our water environment, and this plastic pollutant also can be degraded into smaller pieces of plastic that we call microplastic. This microplastic contamination was found in the water environment, even on the content of bottled drinking water, according to research by Tov et al. In 2019 and Thayer et al. In 2018. Research done by State University of New York at Fredonia, which tests 259 bottles of drinking water from 11 brands. Results obtained are that 93% of water in the bottles test contain microplastics. Especially samples that taken from Indonesia have an average content of microplastic particles around 382 per liter. The dangers of microplastic as pollutants generally can trigger clots in the digestive tract and can also translocate in the human body through the circulatory system if the size is below 150 micrometer. And this is the illustration for the microplastic particle that circulate inside our body if it was contaminate our water. And this is the mechanism of photocatalyst microplastic degradation ability. The degradation ability of microplastic with photocatalyst is depend of the ability of the photocatalyst to generate electron and holes on the valence and conduction band. The mechanism is that the polymer structure will be contact with hydroxyl radical and will be degrade into polymer radical plus water. And for the long mechanism, that will be three intermediate that will be created after the reaction. It's a carbonyl group, aldehyde, carboxylate, and ketones. And last, it will degrade the carbonyl group to CO2 and water. From research done by Ariza Tarazona in 2019, the ability to reduce microplastic is still very low. The research done by Ariza Tarazona used nitrogen doped TiO2 result shown low mass degrade percentage of microplastic pollutant in water. In this research, I use silver doped TiO2 with RGO. Why I use this? According to Ahmed in 2010, the catalytic activity of titania are reliant on the surface and structural properties of the semiconductor, in which including the surface area, crystal composition, particle size, distribution, band gap, and others. And these factors are very important for the degradation. And in 2018, Tafakoli done research to examine the effect of addition of the silver metals and graphene on TiO2 on their photocatalyst performance by assessing the degradation efficiency of the organic acid modeled with acid orange 7. The addition of graphene and silver dope increased the efficiency of degradation of pure TiO2 from 61% to increase to 99%. The experimental method of this research is first I prepare and characterize the catalyst and then go to test the microplastic degradation ability of the catalyst that have been prepared. For the catalyst synthesis using TiO2 P25 ethonic powder as the titanium precursor, while the silver precursor is silver nitrate and RGO used as the source of RGO. The catalyst was first synthesized with PAD method to get silver doped TiO2. And this is the step for the synthesis of silver doped TiO2 and silver doped TiO2 RGO. Next is after the catalyst is synthesized, the catalyst will be characterized with SEM ADX and UV phase. And after the catalyst was characterized, then the degradation of microplastic performance will be tested. And the steps is with 100 millimeter aqueous and 50 milligram microplastic were added in glass, and then 50 milligram of catalyst were added to the glass. The solution was stirred continuously during the degradation test under UV radiation, and the tests were conducted for 1 hour, 2 hours, 3 hours, and 4 hours. The leftover of microplastic after the radiation will be filtered using Wattmann filter paper, and after that, the filter paper will be dried using oven with temperature 100 Celsius for 1 hour. And this is the illustration of the reactor, the photoreactor of our degradation test. The result of semi-ADX characterization of the catalyst, from the figure, we can see that the doped TiO2, it shows spherical particles unevenly sized and a bit of agglomeration occur in certain areas. This agglomeration can occur due to the formation of a solid bridge between surface hydroxyl groups during the drying process, or combination of the particle after being treated with high temperature in the calcination process. In the UV phase characterization, showed that the presence of silver metal dopant acts as a good electron trapper and cause the band gap energy of the catalyst to decrease, and it caused the catalyst to has better ability to absorbing visible light that have wavelength higher than 3080 nanometer, as we can see on the chart. From microplastic degradation, the presence of dopant silver and RGO in the TiO2 gives better ability, better ability in degradation of microplastic. And as we seen in this graphic, the best result that have the higher percentage, the higher degradation percentage of microplastic degradation, it is from the 3% silver doped TiO2 and 1% RGO. This is because the silver dope and RGO has high electron mobility so that the silver dope and RGO can be an electron acceptor and lower the recombination rate of the electron and hole. This given better photocatalyst degradation activity. The conclusion is addition of silver metal dopant and RGO to the TiO2 photocatalyst significantly increase the degradation performance of microplastic in water. The best performance of microplastic degradation is 76%, which is from the 3% silver doped TiO2 and 1% RGO catalyst. And this is the acknowledgement that this study was funded by Beauty Proceeding of University of Indonesia 2020. And I want to also thank all people and organizations that helped me and support the author during this research. And this is the reference for my research. That's all from me. Thank you.
Okay, thank you very much for the nice talk, Mr. Muhammad Haris Fadli. Next, I would like to invite Ms. Lestiani from Center of Applied Nuclear Science and Technology, Batan. Ms. Lestiani will present the characterization of fine particulate matter collected in the vicinity of coal-fired power plants in Java using ADXRF. So, Ms. Lestiani, the time is yours.
Good morning. My name is Dia. I'm a researcher from Center of Applied Nuclear Science and Technology, National Nuclear Energy Agency of Indonesia, Batan. I would like to present one of our research on characterization of fine particulate matter collected in the vicinity of coal-fired power plant in Java using EDSRF. The energy consumption in Indonesia, especially in Java, has risen due to the rapid growth of population, industrial, and economic activities. PLN, Indonesia State-Owned Electricity Company, has a program to build an additional power plant, which is dominated by coal-based power plant to meet this need. Coal power plant has contributed more than 50% to supply the electricity in Indonesia. Indonesia government has a program of 35,000 megawatt of new power capacity which comprises more than 40 power plants, mostly coal-fired type in Java and Bali. This huge and rapid development of coal-fired power plants should be taken into consideration related to the environmental and health impact. The process of coal combustion from coal-fired power plant is significant sources of pollutant since it produces massive amounts of carbon dioxide, sulfur dioxide, nitrogen oxide, and particulate matter. This emission increased the concentration of PM2.5, the particulate matter with diameter less than 2.5 microns, and could travel over hundreds to thousands of kilometers downwind of the plants. Comparison of the PM2.5 with the size of fine beach sand and human hair could be seen in the picture. Divided the human hair into 5 to 7 parts, we'll get the PM10, and divide the PM10 into 4 parts, we'll get the size of PM2.5. Due to its small size, the PM2.5 could penetrate deep into the lung system, and the exposure to PM2.5 has been associated with the cardiopulmonary diseases, lung cancer, and numerous other respiratory illnesses. Therefore, PM2.5 and its heavy metal content should be monitored due to its impact could be harmful to the human health. Nuclear analytical techniques such as EDSRF is very suitable for characterization of airborne particulate matter. EDSRF has been applied to characterize the airborne particulate sample in various pollutant source identification research. There are a limited number of publication-related assessment of environmental impact from the coal-fired power plant in Indonesia with respect to the chemical composition of the fine particulate matter. In this study, we are going to provide the characteristic of the mass, black carbon, and also the elemental concentration of fine particulate matter emitted from the coal-fired power plant. This is the sampling sites. We collected the samples in 4 sampling sites near the coal-fired power plant in Suralaya, Labuan, Cilacap, and Tanjung Jati. The samples were collected using GAN stack filter unit dichotomous sampler that capable for collecting the particulate matter in the PM2.5 to 10, or we call it coarse fraction, and PM2.5 size or fine fraction. Characterization of the samples were carried out for the mass concentration, which is determined by the gravimetric method using the microbalance. The mass concentration were obtained by dividing the gravimetric mass by the volume of the air passing through the filter. The concentration of black carbon in the samples were determined by the reflectance measurement using the EEL smoke stain reflectometer. Elemental analysis was performed using an energy dispersive X-ray fluorescence or EDSRF epsilon 5 from Panalytical. The nuclear analytical technique has several advantages. It has high capability, simultaneous measurement, multi-elemental, high sensitivity, short-time analysis, and it's non-destructive, which make this very ideal for this type of work. EDSRF is an effective tool and allows the characterization of multi-elemental composition and can provide and produce large data sets for source identification. This is the result of PM2.5 and PM10 for each sampling site. The highest concentration of PM2.5 was found in Labuan and the lowest was in Cilacap. Even though this PM2.5 was still below the 24 hours of Indonesian National Air Ambient Standard, but by assuming that these weeks of sampling could represent the annual mean, the PM2.5 in Labuan, Suralaya, and Tanjung Jati would exceed the mean annual of air quality standard of PM2.5. The lowest value of PM2.5 was found in Cilacap. The sample in Cilacap were collected during the coal-fired power plant is still under construction or not yet in operation. Therefore, the PM2.5 was low due there is no emission of coal power plant. On the contrary, the PM10 in Cilacap was higher compared to other sites, since the pollutant source such as road dust or crustal element were higher during the construction activity. This is the result of the PM and BC in 4 sampling sites. Black carbon, or BC, is commonly emitted to the atmosphere due to incomplete combustion. The major source of BC are from the transportation-related emissions such as diesel emission and long-range transport from fossil fuel-related sources such as coal-fired power plant emission, and also biomass burning could be other substantial sources of BC. The average BC contributed 12 to 19% of the PM 2.5 in the 4 sampling sites. This value do not differ significantly with the values reported in the industrial area in Serpong in 2008. The lowest BC was found in Cilacap with similar reason for PM 2.5. And it should be noted that the activity of coal combustion due to the operation of coal-fired power plant in Cilacap was not begun yet. This data could be used as preliminary data or background data before the emission of coal combustion will take place. This is the elemental concentration. Several elements were identified. This table summarizes the elemental concentration of fine particulate matter collected in the 4 sampling sites. It shows that sulfur is dominant and has a highest concentration in the 3 sites of Labuan, Suralaya, and Tanjung Jati. Sulfur in airborne particulate matter is generally present because of the atmospheric conversion of sulfur dioxide to the sulfate particulate phase through the homogeneous process. Hanoi study also reported the high sulfur in PM 2.5 due to the emission of coal power plant. It is worth to be noted that the major emission of the coal power plant was sulfur. The long monitoring to identify the emitted pollutant from coal power plant emission should be considered to monitor the impact of the coal power plant to the air quality and the surrounding environment. In Cilacap site, the major element were the crustal elements such as aluminum, silicon, calcium, and iron, which indicated the road dust or soil sources are dominant. This could be due to the construction activity. This is the calculation of reconstructed mass or RCM. We could define the RCM as a sum from sea salt, ammonium sulfate, soil, smoke, and BC with the given formula. Calculate RCM with a contribution of a total more than 79% for Labuan, Suralaya, and Tanjung Jati, except for Cilacap, because in Cilacap, the major contribution of BC and soil was dominant. This is the conclusion. This study presented the PM 2.5 BC and elemental concentration from the fine particulate matter sample collected in the 4 coal power plant in Java Island. The 24 hours of mass concentration of PM 2.5 was still below the threshold value of 24 hours of national air quality standard in Indonesia, but it has exceeded the annual standard of PM 2.5 except in Cilacap. The elemental analysis of PM 2.5 revealed that sulfur has a high concentration in 3 sites in Labuan, Suralaya, and Tanjung Jati, while in Cilacap, it was dominated by the crustal element. For some sites, the level of elemental concentration was lower than other industrial sites in Java due to the sampling site was carried out before the commissioning or normal operation. Further and periodical monitoring will be needed to obtain a better comprehensive result to assess the impact of coal power plant to the surrounding environment. And this is the acknowledgement that I would like to acknowledge the financial support from the National Nuclear Energy Agency Batam and the Ministry of Environmental and Forestry of Indonesia. And I also would like to acknowledge the help of the staff at the Center of Applied Nuclear Science and Technology for their assistance throughout this study. Thank you.
Okay, thank you very much for Ms. Lestiani for the nice talk. Next, I would like to invite Ms. Sari, also from the Center of Applied Nuclear Science and Technology, Batan. Ms. Sari will present the evaluation of uncertainties measurement in ADXRF for air filter analysis. So, Ms. Sari, the time is yours.
Okay, good afternoon, everyone. Thank you for having me in this opportunity in the 6th International Symposium of Applied Chemistry. It's a pleasure to me to be here to share with all of you in this good opportunity. And now I'm going to talk about the evaluation of uncertainties measurement in EDXRF for air filter analysis. And we have 4 main topics. The first is introduction and experimental method, result and discussion, and the last one is the conclusion. We move to the first topic about introduction. Okay, I work here in Center of Applied Nuclear Science and Technology, National Nuclear Energy Agency, Batan, Bandung. Batan, Bandung have several analytical instruments, and one of them is EDXRF, or Energy Dispersive X-ray Fluorescence, named Epsilon 5 spectrometer. Epsilon 5 usually used to analyze trace elements in filter samples, especially in filter samples, air filter samples, both qualitatively and quantitatively. And then for ensuring the reliability, we have to evaluate the measurement. And then, okay, as we know that to be sure about the data we get from the measurement, we have take several measurements, and every measurement involves an error. And error in the measurement doesn't mean the mistake. So that we need to do several measurements to get a reliable result. Okay. Okay. From the measurements, we get the data, and the data we got from the measurement is the evidence that we obtain from the measurements, and the data can support the theory, while the theory itself is describing the reality. I mean the measurement we did in the laboratory. In this case, the measurement we did with epsilon 5. And in analytical result should always be expressed with its corresponding uncertainty value, so that we need to evaluate the data. And then the next is experimental method. This is the instrument we use in this study, energy dispersive X-ray fluorescence. It's a 3-dimensional polarizing optics. And then the X-ray tubes can be operate up to the maximum power up to 600 watt. And then it has beryllium window, and the maximum voltage is 100 kilovolt with the maximum milliampere up to 24 milliampere. And epsilon 5 also has secondary target because of its three-dimensional. And this is the sample cup. We put our air filter samples on the holder in the sample cup. And then in epsilon 5 also have sample changers, consist of 6 trays and can be filled up to 48 samples. And the first step we have to do is make the application something like a program we use to measure our samples. We name it as APM 2020 because in this year, and the standard using such as slope, intercept, and its correlation. And then we evaluate the uncertainty. And this is the equation we used in this study. The UE5, as you see in the presentation, is expanded uncertainty, and then K is the coverage factor. It's a constant value. It's about more or less is 2 at 95% confidence level. And the E is calibration factor, the slope. This is the uncertainty of net intensity, and this is the uncertainty of blank intensity. And the last is the uncertainty of the calibration factor. Okay, we are going to discuss about the results and discussion. Okay, the table 1 is explain about the optimum excitation condition of the APM 2020 application in epsilon 5 spectrometer. You can see that in this application, we set 5 conditions with different setting and cover several elements in each condition with different energy range. You can see in your view. Okay, and then this is the table 2 about the calibration parameters. After we set the application, then we get these parameters, we get this value, we get the T value, we get the E value, and their correlations. You can see in this page, and then next. Okay, this is the uncertainty estimation. The first is for sodium with the correlation 0.96. You can see this data for sodium. This is the concentration and uncertainty. This is for silica and for sulfur, potassium. This is for calcium, titanium, iron, zinc, bromine, and lead. And this is the table 3. Showed you about the uncertainty value for air filter samples. I mean, this is the ratio for 10 elements, sodium, and so on. This is the concentration range that different from each element. And this is the relative uncertainty. We can see that the value will range from 2 up to 32%. This value then can be used to estimate the uncertainty for air filter samples that measured using epsilon 5 spectrometer. Okay, and the conclusion is from the result I explained, we can see that the epsilon 5 can be used to analyze air filter samples both qualitatively and quantitatively and give the reliable results. From the evaluation, especially for 10 selected elements, were in a good correlation with the percent relative uncertainty will range from 2 up to 32% for sodium, silica, sulfur, potassium, calcium, titanium, iron, zinc, bromine, and lead. Okay, this is the acknowledgement. We would like to extend our thanks to our staff of analysis technique and radiometry team. And then this is the reference, and thank you. Okay, thank you very much for your attention, and please find me afterwards if you have any question. Thank you.
Okay, thank you very much for Amisari for the nice talk. Now I'm going to open the Q&A discussion. Okay, I will check the question. We have some question here. First, the question for Mr. Didit Ardi. First question from Mr. Hafiz. Any other products from microplastic degradation product? Did you analyze generated compound after degrading microplastic? This is for the first question. And the second question from Ms. Wita and Mr. Murianto, also for Mr. Didit. The question is, do you measure the patient degradation by counting the change in concentration? How do you measure the microplastic concentration itself? Thank you. Okay, Mr. Didit, could you answer the question?
Okay, thank you very much. The first question is from Hafiz.
See you the next.
Hello. Okay, we will begin our session shortly. Please, all the participants and all the presenters join this room. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome back to the last parallel session of this conference. And my name is Ilani Putri Ramadhani Tias, and I will serve as your moderator today. Before we begin, I would like to inform you that, as you might already knew, we will have a total of 5 presentations during this session, and we will divide those presentations into 2 parts. The first part will be the presentation from the first 3 presenters, followed by 15 minutes Q&A session. And the second part will be the presentation from the last or 2 remaining presenters, followed by 10 minutes Q&A session. And before I forget, I would like to remind you again that the chat room is only for delivering questions to the ongoing discussion, requesting for changing or moving to the other breakout room, and announcements by the organizing committee. And you can also use a raise hand icon if you want to ask or give us any comments during the Q&A session. So, are the co-hosts ready? Hello, co-host, are you there?
Yeah, ready.
Okay.
It's ready.
Okay. Okay, so we will begin our session with our first presenter, who is Miss Nuryatini, and I think Miss Nuryatini already joined us now. She is from National Standardization Agency of Indonesia, and she will present her work about establishment of traceability chain for pH measurement in Indonesia. Ms. Nuryatini, the floor is yours. Co-host, kindly share the video. Co-host, is the video on? Co-host, we cannot hear the voice.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, in this opportunity, I would like to present about establishment of traceability chain for the pH measurement in Indonesia. Introduction. pH measurement is one of the most common quantitative measurement in many areas, such as this. pH can be used as quality control, which will affect every aspect to the daily life and give impact to decision making. Therefore, accurate, reliable, comparable, and traceable pH measurement is important to prevent duplicating measurement, which will consume more money and time. And this one of the tasks of NMI to provide an accurate traceable measurement to the international system unit. National Meteorology Institute, or NMI, is government institute appointed by national decision to hold and develop national measurement standard. It is responsible for establishment and maintenance of national measurement standard, primary or secondary, and method. Participation in international comparison keep the traceability chain for dissemination of the SI unit to industry and society, ensuring the suitability of the standard for national need, providing meteorological expertise and knowledge to national users, assuring the equivalent of measurement standard, research and development in the field of meteorology for government, industry, and society. History of NMI of Indonesia. In 2004, the NMI, the task of NMI, which was at Kim LIPI, and until 2017. In 2018, task of the NMI moved from LIPI to BSN based on Presidential Decree number 4, 2018. In BSN, NMI function is held by Deputy of National Measurement Standard or Standard Nasional Satuan Ukuran, cover physical, biological, and chemical meteorology. In chemical meteorology, consists of organic, inorganic, electrochemistry, and gas laboratory to provide certificate reference material and disseminate the CRM to the end user to testing laboratory in Indonesia to keep the traceability chain to the SI unit. Electrochemical laboratory establish the traceable chain for the pH measurement in Indonesia through development of the secondary method for pH measurement, production of CRM for buffer solution, scientific testing providing, participation in the K comparison and claiming CMC. This is meteorological traceable for pH measurement. Primary standard as the highest standard of traceability of pH measurement is measured by hand cell as the primary method. The primary standard used to calibration differential potentiometric cell with use for measure secondary standard, and secondary standard used to calibration pH meter for measuring sample. The result of the pH sample is traceable to primary standard. Development of the secondary standard method. In 2015, standard start to development secondary method for measurement as the high traceability chain in Indonesia. This is the system of a secondary standard. It is consists of U-shaped cell which separated by sinter glass. This in the middle of the cell recognize a bulk cell. The bulk cell is immersed in the temperature control water bath. Each cell contains contain identical electrode hydrogen. One cell contains the primary buffer standard solution, then the other cell contains secondary buffer standard solution. The platinum hydrogen electrode then conduct into the multimeter, and the potential difference between two electrodes is measured. The pH of the standard buffer solutions is calculated by this equation. Before routine usage, the method must be validated. The result of validated method for secondary pH measurement of path buffer solution, accuracy and precision of the method was sufficiently satisfying, and estimated uncertainty value is 0.0004. Production of CRM for buffer solution. Buffer solution oxalate, phthalate, phosphate, and borate buffer was measured by pH meter, and phthalate phosphate is measured by meter secondary. The availability of CRM buffer in Indonesia is expected to fulfill the local need, show price import value, and provide the traceability of pH measurement in Indonesia to get accurate, reliable, confident, and traceable results. Proficiency testing is evaluation laboratory performance against pre-established criteria by means of interlaboratory comparison. This is proficiency testing provided by electrochemistry laboratorium. Key comparison are international interlaboratory study used to establish the degree of equivalence between national measurement standards that carried out by NMI. Participation in key comparison for NMI is the best way to demonstrate the NMI confidence in its measurement result and consequently in the measurement capability. Therefore, the measurement result obtained will be internationally comparable and accepted, giving no need to duplicate the measurement in other countries. Electrochemical laboratory has been participated in key comparison PMP QM Q91 in 2015 for phthalate buffer. This measure value is phthalate buffer at 25 degrees Celsius is 4.006 and close to key comparison reference value 4.007. Before claiming the calibration and measurement capability for pH measurement phthalate buffer, we were finished to peer review with reviewer from NMIG. After the peer review, evaluation and document process is required. The action for pH measurement in Indonesia by electrochemistry laboratory is at the beginning level to accommodate the laboratory needs. The electrochemistry laboratory has conducted research activities to build the traceability chain of pH measurement in Indonesia. The activities cover the development of the secondary method for pH measurement, the production of CRM for buffer solution, the organization of pH camp, the participation in key comparison, and finally, the claim of the CMC. Thank you very much.
Adventini, can you hear me? Can you respond to my question? Okay, hello. Okay, hello. Thank you for joining us. So, Miss Natalia will present her work about determination of calcium, natrium, magnesium impurities in demineralized water Bandung 3000 reactors using flame atomic absorption spectrophotometry. Co-host, kindly share the video.
Thank you for this opportunity. We are from Batan, Bandung, would like to deliver our presentation with title, Determination of Calcium, Sodium, Magnesium Impurities in Demineralized Water Bandung 3000 Reactor using Flame Atomic Absorption Spectrophotometry. Low power research reactors such as Bandung 3000 reactor, commonly used in many fields, such as in health, environment, industry, and other applications that provide many benefits to mankind. Bandung 3000 reactor required water which is its function as water coolant, neutron moderator, and radiation shield. Demineralized water used by Bandung 3000 reactor must meet requirements stated in Safety Analysis Report or SAR, such as calcium, sodium, magnesium content, conductivity, and pH water. In this activity, FAAS method was used for determination impurities of calcium, sodium, magnesium in demineralized water 3000 reactor aging in supporting performances of Bandung 3000 reactor to develop science and its application. First step in this activity was preparation, which include sampling of demineralized water from reactor, preparation of working standard solution series, preparation of blank solution for instrument verification, and preparation of spiked demineralized water as method verification. Second step were measurement of standard and sample solution. Quantification was carried out using a standard curve. And the last step were data analysis, which presented not only instrument and method verification, but also the impurities concentration. FAAS instrument parameters we applied were wavelength, slit, lamp current, flame acetylene air, and optimum working range of FAAS. Precision below 1 point. LOD and LOK had been determined also. Method verification was carried out by using spiked demineralized water The result of percent recovery were in accordance with criteria acceptance by AOAC. ORAD values as precision of all elements were in the range between 0.3 up 0.6. According to SAR's requirement, this table presented requirement values of impurities, conductivity, and pH of the demineralized water Bandung 3K 2000 reactor. Concentration of element impurities must below 1 ppm. Conductivity below 3 micromoles per centimeter. Meanwhile, pH water values between 5.5 to 6.5. This table 4, stated that concentration of all elements impurities meets determination impurity of calcium, sodium, and magnesium in demineralized water Bandung 3K 2000 reactor using FAAS gave a good result. Meanwhile, pH water and conductivity gave values were in the range between lower and upper level. This data could be used for assessment and study of Bandung 3K 2000 reactor aging in supporting performances Bandung 3K 2000 reactor. Thank you.
Thank you very much to Ms. Natalia for your talk. And continuing to our third presenter, Mr. Muhayatun. Mr. Muhayatun, are you joining us right now?
Yes.
Oh, yes, I'm sorry, Ms. Muhayatun, I'm sorry. Welcome, Ms. Muhayatun. Thank you for joining us this afternoon. Ms. Muhayatun will present her work entitled Nuclear Analytical Techniques for Identification of Elemental Composition of Fine and Coarse Airborne Particulate Matter collected in Bandung, Indonesia. Ms. Muhayatun, the time is yours. Co-host, please kindly share the video.
National Nuclear Energy Agency of Indonesia. Assalamualaikum warahmatullahi wabarakatuh. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Muhayatun. I work for National Nuclear Energy Agency of Indonesia. Title of my presentation is Nuclear Analytical Technique for Identification of Elemental Composition of Fine and Coarse Airborne Particulate Matter collected in Bandung, Indonesia. As we know that air pollution is a global phenomenon and one of the greatest environmental challenges we face today is air pollution. This is important since more than 50% of the world's megacities were located in Asia, and people live in Asia are the most at risk of ambient air pollution. And compared to other pollutants such as water and soil, air pollution ranked highest in a global burden of disease study. This is air pollution problem in Indonesia. Increased economic development in the Indonesian region has often led to rapid and unplanned urbanization. And urban air pollution is a major problem. You see from this picture that Indonesia has several air pollution problems, including transportation problem in big cities, pollution from industrial activity, forest fire, and volcanic eruption. Therefore, air quality monitoring needs synergy and optimization all the resources we have. And the locus of this research is in Bandung City. This is a Bandung City. And the focus of this research is particulate matter. As we know that particulate matter is an environmental problem related to air quality and public health. And PM can be classified into nanoparticles, ultrafine, fine, coarse, and super coarse according to their aerodynamic diameter. And focus of my research is in here, fine and coarse particulate. Fine particulate is particulate with diameter less than 2.5 micrometer. From this figure you see, the higher PM 2.5, the higher mortality rate. So fine particulates are very dangerous because they can penetrate into the deepest of the lung and heart, causing heart problem, including acute respiratory infection and cardiovascular disease. Why we need fine and coarse PM analysis? First, only few existing data for PM10 in Indonesia, and data on PM 2.5 are relatively scarce. And APM with a mass of less than 1 milligram are very small mass to be analyzed using conventional method. Therefore, nuclear analytical technique, which have high sensitivity, simultaneous, selective, and a very low detection limit are needed. Research that has been done before, this is Air Quality Bandung 2002-2004. The fine and coarse chemical composition have been characterized as well as source enforcement. And long-term monitoring for APM Bandung has also been carried out, and one of the research is used to evaluate the impact of the leaded gasoline ban. As we know, in 2017, Bandung received an award as the city with the cleanest air in ASEAN. So the objective of this research is to provide an overview of the current status of Bandung's air quality, including the level of mass concentration and elemental composition, and its comparison with similar data from 2002 and 2004. And to strengthen the application of nuclear analytical technique for supporting air quality management in Bandung, Indonesia. Methodology, first sampling, and then PM and black carbon measurement, and elemental concentration measurement, and data analysis. Sampling. Sampling will carry out on the office of West Java Provincial EPA using dichotomous sampler chain once a week. And for PM analysis, we use gravimetric method, and black carbon analysis, determination of carbon in the filter is based on the process of light reflection. And black carbon was determined using a digital smoke stain reflectometer. And for elemental analysis, we use EDXRF with 9 secondary targets. This is result and discussion. Table 1. Table 1 is the annual average for PM 2.5, PM 2.5 to 10, and PM 10. And from this table, we see that average 2015 was higher than 2016 and 17 due to the El Nino effect, which caused longer dry season. And compared to 2002-2004 data, mass concentration of fine increased by 49, 27, 35%, while for coarse APM increased by 49, 38, and 6% for 2015, 2016, and 2017. An increase in PM 2.5 level is expected due to the population growth, number of motorized vehicle, and industrial activity. As we know that in national level, the increase in vehicle for 10 years reached 3 to 4 times, and in Bandung also occurs with the same tendency as the national level. This is the black carbon concentration. The black carbon concentration is gentle, which contributes 19% to PM 2.5. Table 2. From this table 2, this is for fine and coarse fraction. This is an elemental concentration. We see that the chemical composition of fine particulate is dominated by sulfur and black carbon. While for the coarse particulate are dominated by crustal elements such as silicon, aluminum, calcium, and iron. And from the correlation and trend, it was show a good correlation and similar trend between these elements, which can be used to estimate the source of the particulate pollutant. A good correlation between sulfur and black carbon can indicate coming from vehicle sources and industrial activity, while the aluminum, silicon, and calcium correlation originate from soil dust and road dust. This is the data comparison in fine particulate during the 2002-2004 and 2015-2017. From this figure, we can see that the increase of sulfur here, okay, can be due to the increasing use of coal, both for power plant and industrial, as well as in vehicle fuels. And the increase of potassium is probably due to open burning activity that are still being carried out in Bandung and its surrounding. An increase of zinc and Mn element here also need attention because these elements are indicator of metal industry pollutant. And reduction of lead concentration can show the success of unleaded gasoline program implemented in Indonesia since July 2006. And this is for the coarse particulate. In the coarse fraction, silicon is a major chemical species, followed by other elements, calcium, aluminum, and iron. And these elements are crustal elements, which generally have a significant contribution to the coarse APM. Okay, this is the conclusion. The annual average of PM 2.5 in Bandung during 2015-2017 is in the range of 17.85 to 20.9 microgram per cubic meter and has exceeded Indonesian standard. Compared 2002-2004 data, PM 2.5 level have increased by 35%, although the annual average of PM10 is still below the standard, but PM 2.5 level that have exceeded the standard and tends to increase from time to time. Therefore, serious tension and appropriate strategy are needed in order to reduce health risks for the community. The characteristic of chemical composition, both in fine and coarse APM, can be used as preliminary study in identifying the source of pollutant, as well as to assess the effectiveness of an air quality program. Acknowledgement, I acknowledge National Nuclear Energy Agency of Indonesia, International Atomic Energy Agency through RAS 7023, Ministry of Environmental and Forestry of Indonesia, and EPA of West Java Province. That's all. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you to Ms. Muhayatun for inspiring talk. Now, before we continue to the next part, I would like to invite all the participants in this room for the Q&A session. You can use a chat message, or you can also use a raise hand icon. I will check first. I see no question both in chat room and in participants. Okay, then I will start with my question. Maybe later or other participants will join me. So I have a question for Mrs. Nuryatini, our first presenter. It's interesting that you talk about the traceability chain of pH measurement in Indonesia. And I saw just a moment ago that your laboratory claimed that the uncertainty measurement for your claim is about 0.004. Am I right? Yeah, and I just want to have like some comments from your side about that claim compared to other countries, maybe like our neighbor country, and how that number affects the uncertainty measurement in our lab, in our routine lab in Indonesia. Maybe you can give us some comments and some.
For the measurement of secondary method, the uncertainty is 0.004 until 0.01. It is suitable.
So is that number like, is it how the written lab in our country? I mean, the written lab, when we measure the pH, how about their uncertainty? Is their uncertainty like below or just in that, so that it's that uncertainty that you claim is sufficient to cover our national requirement?
In NMI, other NMI, for secondary is 0.04 and 0.04.
Oh, okay, 0.04. Yeah, so ours is like lower than other NMI. Okay. Thank you very much for your answer. Okay, I will check again my chat box if there are any questions. Not yet. No question in chat box or no raise hand. Also, okay. Okay, then I will continue with my question then. My second question to Miss Natalia. Miss Natalia, are you there? Miss Natalia, can you hear me? Okay, yeah. Miss Natalia, thank you for your interesting presentation. I would like to ask, before in your slide you present the table and stated the requirement for some parameters like magnesium, sodium, calcium, and pH, and I think conductivity from the SAR. And I would like to know how you determine that that criteria, like the number? You said that the metal should be below than 1 ppm and the pH should be around 5 point something. I'm sorry, I forgot. And then the conductivity, how you set that number? Can you give us enlightenment about that?
Yes, that's the reactor parameter that need from the for the demineralized water requirement that fit to the water reactor needed. It must meet to the SARS requirement.
Is there any like document or reference for there or your lab, how your lab evaluate that number? Is there any reference document for that?
Please give me apologize. Can we use in bahasa?
Oh, yes, sure. Jadi saya mau menanyakan angka-angka yang tadi disampaikan di slide terkait dengan keberterimaan parameter-parameter SAR. Itu kalau boleh tahu angka-angkanya apakah dari dokumen tertentu atau memang lab sebelumnya sudah melakukan evaluasi terkait dengan nilai tersebut, misalnya tadi kan logamnya harus lebih kecil dari 1 ppm, pH-nya.
Iya, baik, angka-angka tersebut sudah ditentukan oleh Badan Pengawas Tenaga Nuklir untuk keselamatan reaktor, Bu.
Baik. Okay. So it's like from our national standard, it's by our country, our standard agency for
our regulation.
Yeah. Okay. Thank you very much for your
information.
Thank you. So there is question in Japanese. So. Oh, okay. Oh yeah, I just read the question. Apologize. There is a question from Ms. Muhayatu to Ms. Yatini. Please, could you explain briefly about the procedures and minimum requirement that must be met for a CMC claim? Ms. Yatini, you may answer that right away. It's yours.
Thank you for your question. For minimum CMC claim, this is the laboratory has a proficiency testing provider. The provider and then production CRM, and then participating in key comparison, and then peer review from other NMI. And then the apply to CM checking.
Okay. Yeah. Thank you. Hello, Ibu. Thank you. Okay, can you explain a little bit more related the peer review? Can we choose the country or they already decided?
We can select the country. Country where the peer review in one expert in
that subject. Yeah. Okay. Yep. Thank you so much, Ibu. Moderator, you still mute?
I am. Okay. Is there any questions? I just want to inform that Miss Dudiana remind me about there is still another presenter, 2 more presenter. Yes, and I want to reply her that we will continue this session. As already mentioned that before we begin the session, we will have two parts. The first part will be the three presenters, and so please still joining with us. And my last question, actually, I would like to ask Ms. Muhayatun. Thank you for your nice presentation. Actually, it's more like a general question. From your results, as you see that the PM 2.5 apparently has exceeded the permitted level of our nationals. Is there any recommendation that you can suggest to our, let's say, our community or our government for how to overcome this finding?
Thank you for the excellent question. We have research related with PM 2.5, not only in Bandung, but in 17 cities in Indonesia, okay? Covering all the big island in Indonesia. So, based on the result, okay, especially in Java Island, most of the big cities, especially the capital city of the province, already exceeded the level, annual level, exceeded the standard. Because the standard is quite low, 15 microgram per cubic meter. Okay. So the problem is how to reduce, okay, to ensure the the environment, clean environment for the community. So first, we have to estimate or to identify the source portion. If we know the source, so the government take action how to reduce the the emission from the sources. So that's the important thing. So I have several publication related to source portion also, but in this conference, I just compare, okay, the situation 2002 and 2017, something like that, and it's clearly so that the PM 2.5 increased more than 35%. So this is the alarm for all of us. That's all. Thank you.
Thank you, Ms. Muhayatun, for answering my question. Okay, I see no more questions and no more raised hands. Then I will close this part. I will continue to the next part. We will still have two presenters. Okay, I will introduce our next presenter, who is Mr. Muhammad Syafiq bin Sulaiman.
Aku nggak ngerti maksudnya si ibu bertanya.
Miss Natalia, could you please mute your Zoom, Miss Natalia? Miss Natalia, hello. Okay, to our next presenter, Mr. Muhammad Syafiq bin Sulaiman. I would like to greet Mr. Muhammad. Mr. Muhammad, are you there with us?
Yes.
Okay, thank you. Thank you. Good evening. Thank you for joining us today. So, Mr. Muhammad is from University Sains, Malaysia, and he will present his work about optimization process for ethylene glycol production using the Pareto solution. Mr. Muhammad, the time is yours. Co-host, kindly share the video.
Assalamualaikum and good evening to all. My name is Muhammad Syafiq bin Sulaiman and I'll be presenting for the paper named Optimization Process for Ethylene Glycol Production using the Pareto solution. Before starting, let's take a look at the outline of the presentation. The first section is the introduction, followed by definition, methodology, result and discussion, and conclusion. So moving on to the introduction, I want to share a picture of a real ethylene glycol. These are the respective abbreviations. Regarding the reactor, the type of reaction that takes place is heterogeneous. The reactions are exothermic and the process is continuous. Now moving on to optimization process. Optimization is utilizing a situation or resource to the best or most effective way. The importance of optimization for this process are energy consumption reduction, maximizing productivity, reduction of side products, and minimizing feed usage. For this paper, optimization is performed on the reactor for maximizing production and minimizing energy usage. However, both of these parameters are in conflict with one another. This is the problem faced. This can be solved by using multi-objective optimization approach using Pareto solution. This is the objective. Next, I'm going to lay out the problem statement and objective of this paper. For problem statement, the concerns are on balancing conversion, and energy consumption, and to solve the contradictory objective. For objective, it focuses on process optimization and multi-objective optimization. Now for the second section. Multi-objective optimization is defined as an area of multiple criteria decision making, with more than one objective function being optimized simultaneously in the presence of trade-off. For Pareto solution, it is a method where non-dominated solution sets are collected and boundary points are defined to form Pareto optimal front. From that, Pareto optimal set is identified, followed by the calculations to obtain Pareto optimal solution. Into the third section. The flow of methodology starts with reactor modeling, followed by objective correlation, objective optimization, and multiple objective optimization. For reactor modeling, a model of Yu and Chen from 2017 is taken. The reactor is a plug flow reactor with the following parameters. Next, the objectives are correlated with the equation of Q equal to mc theta. From this, some derivations are made to make the objective be the subject. In optimization, two objective optimization, also known as single objective optimizations, are performed, named low energy and high product optimization. Low energy is minimizing energy consumption, while high product is maximizing ethylene glycol production. From the objective optimization, Pareto front is obtained. From the Pareto front serves as boundary for the multi-objective optimization. The multi-objective optimization uses E-constraint method to gain the Pareto optimal solution. The E-constraint method is done by keeping one objective and restricting the rest. The following are the equations and their respective abbreviations. Now moving on to result and discussion. For objective optimization, which refers to low energy and high product optimization, ethylene glycol is objective 1 and hydrogen flow rate is manipulated variable 1. Based on this statement, the slow increase is caused by maximum reaction rate limit. The graph shows objective 1 against temperature, which is manipulated variable 2. The reason for this statement is due to more side product form as temperature increases, which follow Le Chatelier's principle. On with the third graph, which shows objective 2, which is energy consumption against manipulated variable 1. The statement points that the graph obeys the law of thermodynamics. The fourth graph shows objective 2 against manipulated variable 2. A better explanation is that at first, the energy released from exothermic reaction supply additional energy, but as the temperature increases, the energy requirement shoots as well. For multiple objective optimization, the optimum value for ethylene glycol production and energy consumption is as shown. The trade-off made for both are also shown. The graph shows the Pareto solution. Point B is selected as the optimum point for the multi-objective optimization. On with the comparison of single and multi-objective optimization. It can be seen that there's a huge difference in terms of energy consumption, with objective optimization having lower value. Multi-objective optimization best single objective optimization in terms of reactor temperature, but it's the other way around for ethylene glycol production. The high amount of hydrogen gas used for multi-objective optimization can be managed by proper recycling, in which reduces wastage. In conclusion, multi-objective optimization approach with a constraint method has been applied to find the Pareto optimal solution. The interaction between manipulated variable 1 and variable 2 with objective 1 and objective 2 is highly dependent.
Okay, thank you, Mr. Muhammad, for the nice presentation. Heading on to our last presenter, who is Ms. Nurhani Ariana. Ms. Nurhani Ariana, are you joining us? Okay, thank you very much for joining us today. And Ms. Nurhani Ariana is from Research Center for Chemistry, Indonesian Institute of Sciences, and she will present her work entitled Proficiency Testing for Determination of Pesticide Residues in Black Sea, Comparison of Three Robust Statistical Approach based on ISO 13528 to estimate the consensus values for a small number of participants. Co-host, please kindly share the video.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Nurhani Ariana from Research Center for Chemistry LIPI. Thank you very much for the opportunity for me to share our article entitled Proficiency Testing for Determination of Pesticide Residues in Black Tea, Comparison of Three Robust Statistical Approaches based on ISO 13528 to estimate the consensus values for a small number of participants. So for the first slide, the introduction. So definition of proficiency testing is evaluation of participant performance against pre-established criteria by means of interlaboratory comparisons. So this is the general proficiency testing SIM mechanism. So in PT SIM, there are PT provider and the PT participants. In this case, the PT provider is RC Chem LIPI, and the PT participants are testing laboratories. The PT provider in the SIM will distribute the PT items to the PT participants. And then the PT participant will analyze the PT samples and then submit the PT results back to the PT provider. And then the PT provider will perform statistical data evaluation, calculation of performance score, and publish the PT report. In this case, the objective of the PT SIM was to determine the concentration of pesticide residues in black tea matrix. There are 11 participating laboratories from Indonesia, Myanmar, and India. And to calculate the performance score, we use an assigned value. The assigned value, according to ISO 13528, can be determined by several ways, but the two most used assigned values were reference assigned values and consensus value from participants' results. In the real PT sim, we use the reference assigned value for calculation of performance score. However, in Indonesia, many PT sims were still operated by the consensus values derived from the participant results. The challenge is when the PT sim was only participated by a small number of participants and the test results were highly varied. The performance evaluation by using consensus values from participant results may become unreliable. In these conditions, many PT providers were not able to perform the statistical data evaluation, causing the needed PT become unavailable. Therefore, our objectives in this study was to investigate some data treatment processes for PT with small number of participants, including to find suitable outlier identification methods and to compare 3 robust statistical approaches recommended by ISO 13528 for calculating consensus values from participants' results, which is variance of algorithm A, Hempel method, and M estimator. So this is the method. The participants' data used in this study were taken from PT sims for determination of pesticide residue, endosulfan sulfate, and bifenthrin and black tea matrix organized by Tentang LIPI in 2014 and 2015. All of the statistical data evaluation were done in accordance with ISO IEC 17043 and ISO 13528 by using the Microsoft Excel. There are 3 steps. The first step is outlier identification. We use 3 methods, the graph test, Hempel test, and trim average. And then the second step is estimation of the consensus values. We use variance of algorithm A, Hempel estimator, and M estimator. And then the third step is the performance evaluation. We can use the Z-score or the Z-ascent score. The detailed calculation will not be shown in this presentation due to the limited time. And here is the result. From the total 11 registered participating laboratories, there were 8 laboratories submit the result for endosulfan sulfate, and 10 laboratories submit the results for bifenthrin. In the PT with small number of participants, information about the distribution patterns of the data, such as normality, is needed before applying any statistical analysis tools, since typical statistical tests incorporate assumption about the underlying distribution of data. Firstly, we made the histogram of the data to assess the distribution of the data and detect visually of any outliers. The histograms are factory, especially when many outliers may be present. Therefore, in the study, we compared 3 outlier identification methods. The graph test and the Hempel test compared the difference between an extreme value and the center of the data with the variability of the data and identify the extreme value as an outlier if the ratio is too large. Now, in this case, however, both of these tests cannot identify the outlier in the data set. Therefore, in the table, the results for graph test and Hempel test were all okay. The outlier finally can be detected by the trim average method. And the third one, this is the comparison between the obtained consensus values of participant results with the homogeneity test results derived by GCECD measurement and the stability monitoring results derived from GCIDMS measurements and the assigned value from the real PT sims. For endosulfan sulfate parameter, the consensus values of participants' results obtained from 3 robust statistics were inside the range of tolerance, and close to the other results, the homogeneity test, the stability monitoring, they were close. However, for bifenthrin, the obtained consensus values were close to the low limit of the range of tolerance. This may be caused by some technical issues in the sample preparation, maybe an analysis of the target analyte. And finally, the calculation of the performance score. All of the three robust statistical approaches can be used to evaluate the PT results and were able to detect the acceptable warning and action signals. For endosulfan sulfate, the highest percentage of acceptable Z-action score was achieved by the HEMPA method. The consensus value of endosulfan sulfate obtained by the HEMPA method was also the closest to the other results. And for bifenthrin, the three robust statistical approaches give the same percentage of satisfactory, questionable, and unsatisfactory results. The consensus value of bifenthrin obtained by the M estimator was the closest to the homogeneity test results and stability monitoring results and the assigned value from the real PTCs. So the conclusion in this study, the three robust statistical approaches recommended by ISO 13528, which is the variance of algorithm A, HEMPA method, and M estimator, were successful in estimation the consensus values from participants' results and were able to assess the performance of participants. And the most adequate statistical approaches to the closest consensus values compared to the homogeneity test results, stability monitoring results, and assigned value in the real PTCs were HEMPA method for endosulfan sulfate and bifenthrin for and M estimator for bifenthrin. The highest effectiveness in achieving the acceptable Z-action score for endosulfan sulfate was achieved by the HEMPA method. Meanwhile, for bifenthrin, all of the robust statistical approaches gave same effectiveness in assessing the performance of participants. That's all my presentation. Thank you very much for your attention. Good afternoon.
Thank you very much, Mrs. Nurhani Ariana, for nice presentation. Now, I would like to invite all participants to join us in the Q&A session. Please write your questions or your comment in the chat box. I see no chat box. I see no raised hand. Okay, maybe I will start with my question then. I have a question to Mr. Muhammad. There are really nice presentation. I would like to know if the Pareto solution, as you described earlier, is there any like bias? How accurate you can determine that process using your solution, or is there any like drawback or any something that we need to take into account during the process when we want to evaluate the process using the Pareto solution? Can you give us some enlightenment?
I get your point.
Yeah.
So in terms of drawbacks, there are some, but that depends on the amount of data you gathered. So the higher amount will tends to be more precise in terms of data gathering, so that you don't miss any crucial points, any points that deviate from the actual or theoretical state. So yeah, that's all.
Okay, thank you. Thank you very much. Thank you very much for your answer. Okay, I will check again my chat box online. No other question. Okay. Is there any question? Is there any participant that want to raise their hand? You can use your raise hand icon.
My question in the chat box, Ms. Dylan.
Oh, okay. Sorry. I will. Okay, there is a question from Miss Dia from Batam to Miss Pulhani. Could you explain briefly what is the preparation of PT samples and how do you get the value? Thank you. Miss Pulhani, you may answer the question. The floor is yours.
Thank you for the question, Miss Dia from Batam. In the preparation of PT samples, first we bought the commercial tea, commercial black tea, and then we grind it, and then we filter it until the desired size of material. We choose about raw material. We prepare about 2 kilograms of PT samples at that moment. So the final concentration is hopefully in 400 microgram per kilograms, and for endosulfan sulfate, and about 800 micrograms per kilograms for bifenthrin. So in that moment, in the real PT sims, we use the GCMS to assign the reference value. But in this study, we try to search for the suitable approach for the consensus value. That's my answer. Thank you.
Okay, thank you. Okay, thank you. So you use the PT SAM using the consensus value, right?
Yeah, in the study.
Okay, thank you.
Okay. If there are no more questions, I will end this session then. Okay.
I have a question in the chat box.
Okay. Oh, okay. There is a question from Ms. Muhayatun Batan from Ms. Norhani. Could you please explain why only a few participants took part in this PT? Is there a minimum criteria for participants? Ms. Norhani, you may answer the question. The floor is yours.
Yeah, thank you very much for question from Ms. Muhayatun from Batan. I will answer. So why only a few participants took part in this PT? So in that moment, in 2013, 2014, the network for organic metrology laboratory is not, there's no much organic laboratory in Indonesia. Yeah, we still develop in that in 2013. So although we already published the PT SIMS, there are only about 3 laboratories from Indonesia join in the PT. And then in the next year, we published the PT SIMS to the countries in Asian region, and then about 8 more laboratories join the PT SIMS. So we use the exact sample in both PT SIMS. So in that moment in 2013, our network is organic metrology network is still limited. And for the minimum criteria for participants for to try a lot of some statistical data evaluation. So for consensus value from the ISO 13528, the minimum participant is recommend is about 20 participants. Thank you very much.
Ms. Wayatun, is there answer your question?
Yeah, thank you so much.
Okay, thank you. Oh, I think our session is already finished due to the time constraint. I would like to extend my gratitude to all the presenters for their inspiring talk, and also to all the participants for joining us in this room today. Thank you very much. And before I forget, I would like to remind you that after this session closes, you will be asked to join the plenary meeting for the closing remarks and also the maybe some announcements from the committee organizer. So please be joining our plenary meeting after this session. So I apologize for any inconvenience during this session. Thank you very much for your attention. Stay safe. Wassalamualaikum warahmatullahi wabarakatuh. Thank you. Good afternoon, everyone. Good evening. Good evening, everyone.
Thank you.
Thank you, Dila.
Thank you. Thank you. Have a good day. Thank you. Thank you, everyone. You may leave the room and join the plenary meeting. Thank you. To all participants, I would like to remind you to join the plenary meeting for the closing ceremony. For all the participants, I would like to inform you to join our plenary meeting for closing ceremony.
Good afternoon, ladies and gentlemen. We really appreciate for your participation until closing ceremony. Our honorable speakers, moderators, participants. Finally, we come to closing ceremony of the ISAC 2020. We ask the Director of Research Center for Chemistry for closing remarks. Dr. Reni Meliana, the time is yours.
Thank you, Marisa. Assalamualaikum and good afternoon, ladies and gentlemen. Alhamdulillah, I believe that during these 2 days, all of us have obtained a fruitful discussion in this 6th ISAC 2020, which main theme is natural medicine and nutraceutical. We have learned a lot from the keynote speaker delivered by our keynote speaker, Professor Gunawan Indrayanto, thank you, Dr. Ambara Rahmat Pradipta, presenter, and participant who have joined this conference. I certain that every presenter have presented their own research. I sincerely hope that through your presentation and discussion afterwards, all of us can contribute for better research in the future, and we can create good collaboration among us. Finally, I would like thanks to all the committee members for all this hard work organizing this conference and ensuring that it processed smoothly. I would like also to apologize for any inconvenience that you might experience during this conference. Ladies and gentlemen, all the participants and honorable speakers, once again, I thank you for your participation and wish you all stay healthy. See you again in the next 7 ISAC 2021. Thank you, Marisa.
Thank you, Dr. Yeni, for the officially closing our symposium. Ladies and gentlemen, and all plenary speakers, it's time for us to open our video. Maybe the committee will take the last documentation for today. Okay, thank you for the open the video. Also, we'd like to say thank you for spending the time with us, for all participants and also plenary speakers. And also, last but not least, we hope we can meet on the 7th International Symposium of Applied Chemistry 2021. I'm Marisa Angelina. As a master ceremony of the symposium, would like to apologize for the inconvenience during our symposium. Good afternoon, have a nice day, and see you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you all.
Bye. Thank you.